welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner.
All right. Well, welcome everybody to our day 2 of the Generic Drugs Forum 2025. Good afternoon, good morning, or good night, depending on wherever you may be. It's wonderful to see so many people in person and online with us today. We certainly appreciate your commitment in attending this event wherever you may be across the globe, and also overcoming any scheduling challenges. If you are joining us in person and didn't get a chance to see it yesterday, please note the online Wi-Fi login information here on the slide, and it's also posted on the registration desk. I've also seen it posted on some of these pillars, so you might look around and see that there as well. My name is Corey Adair, and I am with the Small Business and Industry Assistance, or SBIA program, which resides within the Division of Drug Information, or DDI, and the Office of Communications, or OCOM, within CDER at FDA. It is a great pleasure, along with my colleagues from the Offices of Generic Drugs, Pharmaceutical Quality and Translational Sciences, to be with you here today. We implore you to make the most of this experience, to interact with the subject matter experts, and attend all of the sessions that interest you. Learn all that you can, submit your questions, and network in person as well as online. We are also offering YouTube live streaming, so please go ahead and take a moment to invite your colleagues. Now, here are some overarching learning objectives for today, which are applicable to both conference days. Now, let's turn our attention to some logistics as applied to all attendees. First, here is the link to our one-stop shop for answers to your technical questions, for downloading the files, accessing the survey link to obtain the certificate of attendance, and so much more. For online attendees, this is also the same page that you used this morning to access the event. So please bookmark this page as you will need it over the next 2 days. If you have any technical questions about this conference, please check there first and you will most likely find the solution. Next, on our one-stop shop page, there are session evaluation links and also our final evaluation and survey, which will be activated this afternoon. And this link will also be sent in our post-event thank you email within 24 hours of the conclusion of today's event. Upon completion of this survey, an attendee can then download a certificate of attendance, which can then be used in support of RAPS, SOCRA, ACRP, and also SQA credits. The trick is that this certificate must be downloaded immediately after completion of the survey, otherwise that opportunity is lost. Even if you do not want or need a certificate, please take a few moments to complete the conference evaluation, as your feedback will help guide the design of our future events. As Brenda mentioned yesterday, we do have one key new question regarding our SBIA Learning Repository, which contains over 800 webcasts curated from previous SBIA conferences and webinars. Your response to that question is invaluable for our planning. That question, as a reminder, is, do you believe that a comprehensive, discipline-based, and difficulty-ranked generic review forum or workshop curriculum aligned with the FDA's generic drug review process would improve knowledge retention, enhance regulatory understanding, and better prepare industry professionals? Again, we greatly appreciate your response to this question, so please take a moment when completing that survey and evaluation to answer that question for us with a detailed response. And so here we have here. Now, please note that the evaluation and certificate are only available for 2 weeks following this event, which means that deadline is April 24th. Also, we did briefly advertise for a time that this event would offer continuing education credits for physicians, nurses, and pharmacists, but please note that we can no longer support these continuing education credits. Also, as a reminder, we are live streaming and the YouTube links will be posted to our website post-event. That website link is www.fda.gov/CDER-SBIA. In the interim, the YouTube links for both days will be posted on our one-stop shop link. That is that first bullet point that you see here on the screen. All right. And so additionally, if you're online, for our online attendees, make sure that you submit your questions using the on-screen link or the QR code. Some additional details for our in-person attendees as well. Like I mentioned before, please note the on-site Wi-Fi. It's on here on the screen as well as on some pillars or the registration desk if you need. For the QA, many of you were here yesterday. I'm seeing some familiar faces in the audience, but as a reminder, please line up at the 2 microphones when the Q&A time comes, and that way we're aware of what questions are coming up, and we can balance the online questions as well. As a reminder right now, just take a moment, please silence your phones, silence all your laptops and anything else. Just make sure those notifications are turned off. And additionally, please use the internet sparingly while we're here. It may impact our online delivery of our content materials today, so we appreciate you using discretion with that. Additionally, you are on your own for lunch here. There is the Corby Kitchen in-house on the first floor. There's also several eating establishments nearby in the vicinity within walking distance. So you can get more information at the registration desk if you'd like a full comprehensive list of what resources and lunch options are available nearby to you. And lastly, I also did want to note that we do have some printouts of relevant guidance documents for our in-person attendees outside. They have QR codes, so you can use your phone to scan those, and they will take you to ones that are relevant for today's presentations. So next, my co-host for today is Brenda Stodart, captain in the U.S. Public Health System, and she's also the director of SBIA within DDI and OCOM as well. And now it's time to begin today's agenda. As a reminder, all of our speakers' bios and headshots are available on our One Stop Shop website if you'd like to learn a little bit more about them. But for our first sessions, first we will hear from Ravikant Kona, who is a pharmaceutical scientist in the Division of Quality Assessment 1 within the Office of Product Quality Assessment, OPQA 1, and the Office of Pharmaceutical Quality, or OPQ, within CEDAR. Next, we will hear from Reynolds Ray Contave, or Dr. Ray Contave, and Senior Regulatory Health Project Manager in the Enterprise Project Management Staff within the Office of Quality Assurance, or OQA, and OPQ at CEDAR. And then next, we'll hear from Dr. Paramjit Kaur, a team leader in the Division of Bioequivalence 2 within the Office of Bioequivalence within the Office of Generic Drugs within CEDAR. So at this time, please join me in welcoming Dr. Ravikant Kona.
Thank you, Corey, for the introduction. Good morning, everyone. I want to thank you all for being here in person and those connect remotely. My name is Ravikant Kona. I am a drug product assessor in OPQ. The title of my presentation is General Quality Considerations of Drug Product Label for Alternate Dosing Administration. As the title indicates, I will provide a high-level overview of quality considerations. So what I wish to accomplish from this talk is hopefully by the end of this presentation, I want the audience to take away the following. Understand the general quality considerations of the drug product labeled for alternate dosing administration. Identify the relevant FDA guidances. And learn effective data submission for products labeled for alternate dosing administration. In the outline of talk, I will discuss what is an alternate dosing administration and the common types. I will also discuss quality information needed to support oral label use. Along the way, I will present a few case studies and considerations, and I will finish my talk with challenge questions. So what is an alternate dosing administration? So it refers to a method of delivering a drug or medication through a different route other than the standard or conventional one in order to achieve the desired therapeutic effect. So my talk will primarily focus on these three commonly used use of soft food or liquid as vehicles, enteral feeding tubes, as well as admixtures. So we'll start with the first one, use of liquids and or solid food as vehicles. I want to refer to the draft guidance. It's in the bottom of the slide. It provides recommendation for selection of vehicle and as well as the in vitro test to qualify a vehicle. Image onto your right is taken from the guidance, which lists all different soft foods and their approximate pH ranges. And I think it's critical to understand the pH range of the vehicle that you select, because it's going to, for example, if there is an entry-coated tablets or pellets, choosing a appropriate vehicle would impact the drug release. So it's probably going to strip off any entry-coated pellets and thereby altering the pharmacokinetic behavior. I want to specifically refer to the footnote 4 of this guidance. And this is applicable to the generic generic products. So it says the vehicle studies on the RLD would establish the compatibility of the active ingredient and need not be repeated unless there is a risk that the formulation of the ANDA would have a different impact. So in that case, the recommendations provided in this guidance are useful. So what are the general considerations for selection of vehicle? Compatibility and suitability. The guidance recommends that the applicant consider properties of the vehicle, such as acidity, alkalinity, chelating or binding effects, especially for entry-coated tablets. pH higher than 5.5 would impact the coating behavior and alter the drug release. Suitability, the applicant is recommended to evaluate flavor, texture, mouthfeel. Any graininess can trigger chewing, again altering the drug release properties. Impact of vehicle, the applicant is recommended to evaluate the impact of vehicle on potency and drug release. Palatability and swallowability, acceptance in terms of taste, aftertaste, smell, texture. Swallowability, patient should be able to take without gagging or choking. In cases for repeat administration or multiple use, it is recommended that applicant evaluate in-use stability data as well as microbial assessment. And in special cases where vehicle mixture is administered along with the feeding tubes, I will discuss the in vitro tests in my subsequent slides. So method of assessing the impact of vehicle on the product quality attributes. Obviously, you need to have a robust analytical method. It should be developed and validated. Any source of interference should be determined if needed. Now, the testing, this guidance discusses evaluating the integrity. Integrity is usually not an issue if you are administering the whole drug product, but if you are manipulating the drug product, say if you are crushing, then any changes to the polymorphism or loss of bead integrity should be assessed. For potency and stability, for immediate administration, it is recommended that study be conducted a 2-hour incubation period, and as well as the homogeneity. So I want to point out in the guidance that if the RLD label doesn't indicate the incubation period, the guidance recommends that you perform the studies after 2 hours incubation. Now, for the drug product labels for sprinkles, so sprinkle formulations are drug-containing pellets or granules that can be mixed with soft food before administering. The image I have here on the right side, it shows different ways a drug product can be administered in the sprinkle formulation. It could be in intact, like granules, pellets, powders, or beads, or it could be manipulated, like crushing a tablet, emptying a capsule content, or making serial dilutions, or mixing a syrup into a vehicle. Considerations for sprinkle, obviously the bead size is important. The guidance I've referenced in the bottom of the slide recommends a target bead size of 2.5 mm. to a maximum size of 2.8. And bead size is recommended to be performed as per USP 786. And if the applicant choose to provide a bead size greater than the recommended, then adequate justification should be included. Now, there are times where the sprinkles are also administered along with the enteral feeding tube, in which case all the beads are able to safely pass through the feeding tube and not cause any tube occlusions. And the in vitro tests also need to be performed. Now, I want to go on to the case study 1. This is an immediate release capsule formulation. It's a sprinkle. You will notice here the label does not specify the softwood. Also, it's because it's an immediate release. It's an immediate use, so it doesn't specify any incubation period as well. The applicant do not provide any comparability study. Agency issued a deficiency to address the label use. So orally performed tests. So these are cellulose-coated pellets. They are not sensitive to pH. They are neutral in taste, so there is no risk for patient compliance. So the applicant evaluated different food here. If you look at the soft food that they have choose, ranges the pH range anywhere from 3 to 8. And as per the guidance, the applicant provided comparability, suitability, impact of the vehicle, palatability, and as well as the incubation up to 2 hours as per the guidance. So the information was found to be adequate. And this is all the testing data that they were provided. Another case study here, you'll see the label indicate the use of 3 different vehicles: cherry syrup, sugar-free Aura Sweet, and simple syrup. And here's the storage conditions. So when the applicant provided the data, the stability data show an unknown impurity when mixed with cherry and simple syrup that was exceeding the ICH limits. The applicant did not provide any qualification data of the unknown impurity, so we issued a deficiency to provide identification and qualification information. So in this case, the API was interacting with the reducing sugars and forming an adduct. So that was showing as an impurity. We will move on to the enteral feeding tubes. A feeding tube is a medical device used to provide nutrition or medication to patients who cannot obtain a nutrition or medication by swallowing. Based on the site of insertion, we have different enteral feeding tubes. NG tubes, G tubes are the most common, and they vary by tube diameter, tube length, composition, number of ports, connectors, geometry, and eyes. And I have an image on the right side to show the different feeding tubes. So I just want to mention that the variability associated with the insertion point and final sites of drug release are outside the scope of the quality review. So what are the considerations for generic drug products? The applicant should demonstrate the drug product is therapeutically equivalent to RLD when administered according to the label use. Instructions are provided in dosage and administrative section of labeling. And testing information can be found in the RLD packaging insert or product-specific guidance. And testing conditions include number of units, tube type, size, brand, and the size of syringe used. If the labeling instruction specifies immediate administration after preparation, the agency recommends performing comparative in vitro studies at both 0 and 15 minutes soaking times. Now, what are the in vitro testing? Recovery. So the applicant should evaluate the recovery. It's the percent of the drug of the dose before and after administration. Sedimentation volume and redispersibility. Any insoluble components tend to settle and clog the tube. So the applicant is recommended that they perform sedimentation volume studies and redispersibility. If the solid settles, the applicant should make sure that it can be redispersible before administration. In use study, the applicant should demonstrate that the drug product dispersion is chemically and physically stable. Particle size distribution, acid resistance is in the percent drug release in acid media, and the dissolution. So I want to move on to a next case study. This is an immediate release tablet formulation. Applicant provided data using one material, only 8 French and 12 French. Per the guidance, the FDA recommends the applicant test at least 3 different dental tubing configuration for the tubes. So we issued a deficiency. The applicant came back and provided the data as follows. And the testing was performed at 0 and 4 hours as per the RLD label. So here's another case study of granules that was building up. In this example, the product-specific guidance recommends the use of NG tube 8 French with 8.5 pH as a worst case. The applicant evaluated as per the guidance. However, a granule buildup was observed and no buildup was seen in RLDs. So we requested the samples. Samples were sent to the FDA lab, which showed large number of granules retained in syringe, and very low recovery was observed in ANDA compared to RLD. And the applicant wanted to, based on the data, applicant wanted to include additional instructions which are different from RLD label, and the product cannot meet the RLD label for alternate administration, which is inadequate measure. So we recommended reformulating it, and the applicant came back, reformulated the product to address the sticking of the pellets inside the syringe. So the next is drug product administered as admixtures. A pharmaceutical admixture consists of a drug product mixed with an appropriate diluent in a suitable delivery device for the purpose of parenteral infusion. ICH Q8R2 section F recommends that applicant perform a comparability or compatibility studies for the reconstitution diluents. Commonly used diluents: saline, 5% dextrose, ringers, lactated ringers. Now here's a case study that I wanted to show where the applicant provided all the information, and we ended up not issuing any deficiency because all the information provided was as adequate. So this one, this product can be reconstituted with water for injection, or also it can be administered intravenously. And the data the applicant provided supports the RLD label use. In raw non-refrigerated water, the applicant provided up to 1 hour at room temperature and 8 hours at refrigerated condition. For IV administration, the applicant reconstituted using sodium chloride and lactated ringer per the RLD label. and the applicant provided appearance, pH, color, clarity, assay, osmolarity, and visible particles. So I want to move on to the challenge questions now. Testing recommendation for in vitro feeding tube study include recovery testing, sedimentation volume and redispersibility testing, particle size distribution study, all of the above. I'll give you a few seconds to think about it. How many of you think it's A, B, C, D? That's right, answer is D. Which of the following statement is not true? Sprinkle formulations are drug product pellets or granules that can be mixed with soft food before administration. Compatibility study with soft food to support orally label is not a requirement. Agency size of 2.8 mm. Assessment of drug substance in the mixture includes integrity, potency, stability, and homogeneity. How many of you think A, B? That's right, it's B. So in summary, we looked at different drug product quality considerations for dosage form labeled for alternate administration, discussed several case studies along with quality information necessary to ensure the drug product meets RLD label requirements. And hopefully this information provided helps sponsors submit effective data, resulting in fewer review cycles. I want to acknowledge my colleagues at OPQA, Dr. Wu, Dr. Oliver, Dr. Pottenker, Dr. Nguyen, and I will also like to thank GDF Planning Committee for giving this opportunity. I will leave with these resources, and I will be back to answer your questions. Now I'll hand it over to Ray.
All right. Good morning, everyone. I'm Ray Kantav, Dr. Ray Kantav. I'm a senior project manager sitting in the Office of Pharmaceutical Quality. It's my pleasure to be here with you today. Thank you for having me. My talk will cover nitrosamine guidance, which is a topic that seems to come up quite often nowadays. So let's get started with some learning objectives. What do I hope to accomplish today in this presentation? Today, I aim to clarify differences between two structural classes of nitrosamine impurities, discuss the origin and the purpose of the carcinogenic potency categorization approach, or CPCA, introduced in September of 2023, outline the elements of the three-step mitigation strategy, highlight key updates from the 2024 nitrosamine guidance, and if time permits, I hope to touch on a few other ideas as well. Now for a brief history or historical background. In June of 2018, FDA was alerted to an impurity issue impacting valsartan products that led to voluntary recalls in industry, and within CEDAR, the formation of a rapid response incident management group tasked to take, in part, quick action to identify the root cause of this issue. 8 years later, I can tell you that rapid and quickly can be quite relative. However, our efforts were not in vain. With the discovery of NDMA and valsartan products, global regulatory agencies and pharmaceutical managers worldwide sprang into action. FDA initiated collaboration with international regulatory partners, seeking alignment on acceptable intake limits, or AIs, and testing methodologies when possible. We also shared best practices and assessment strategies through international working groups, and later participated in global initiatives for nitrosamine control, such as Health and Environmental Science Institute, or the HESI effort, which is going on today. This is because the impurity issue is not isolated to NDMA and valsartan. Within a few months, we saw the scope expand rapidly to include NDEA and valsartan, NMBA and losartan, NDMA and ranitidine and metformin, CPNP and rifampin, the gift that seems to keep on giving. Today, our investigations continue, mostly focused on NDSRIs in various products. With that introduction, let's take a step back for some background. I hope this is helpful to orient all in the audience, knowing that some of you may be more familiar than others. Fortunately for you, organic chemistry is not my strong subject, so I can keep this very, very high level. So, what are nitrosamines? Simply put, they are chemical compounds where a nitrosyl group is bonded to a susceptible amine. Since the 1970s, research has shown that these compounds are genotoxic and probable human carcinogens. Though nitrosamines can be naturally occurring in the environment and can be found in foods, they can also be synthetically manufactured in pharmaceuticals and also in rubber. So on to the structural classes. Initially, we identified small molecule nitrosamines like N-nitrosodimethylamine, or NDMA, which are primarily formed as process impurities from secondary reactions in drug substance synthesis. We will find this chemical structure of NDMA here on the left side. We have called NDMA and impurities like it small molecule nitrosamines because of their relatively low molecular weight. Later, we discovered a different type of nitrosamine drug substance related impurities, which occur primarily in drug product manufacturing when nitrosating agents, often coming from nitrite-rich excipients, react with susceptible amines within the drug substance molecule, or the API molecule itself, leading to formation of nitrosamine of a relatively higher molecular weight. You will find an example of that on the right, where a nitrosyl group, which is circled, bonded to the chemical structure of phenylephrine. So that's the API structure itself. So with a surge of voluntary recalls and new nitrosamine signals, there was an increasing public interest in the impact of nitrosamines that they might have on the nation's drug supply. This prompted FDA to communicate our understandings and recommendations through guidance documents. Our lab in St. Louis played a crucial role in our effort to respond to the nitrosamine issue. As they worked through challenges in development of analytical methods used to test material like drug product or drug substance, we gained insights that shaped our guidance. As our chemistry assessors worked through issues when evaluating method validation reports, we observed sensitivity, specificity, and other method-related deficiencies. When evaluating root cause investigations submitted, we observed risks associated with raw materials, degradation reaction pathways, manufacturing conditions, recycled solvents, and even packaging. This is a big kudos to the industry for doing their research and providing us as an agency with this information that was critical to our guidance. We believe that sharing those insights will be helpful to manufacturers in their testing and mitigation efforts. As new nitrosamines were discovered, our toxicologists found it increasingly challenging to derive corresponding acceptable intake limits, or AIs, due to the lack of available, robust, compound-specific carcinogenicity data for these impurities. So importantly, we wanted to ensure that we could communicate our current understandings on AIs for nitrosamines, as this was essential for setting targets when manufacturers were doing their sample testing. Our colleagues in industry to our colleagues in industry, nitrosamine guidance would communicate FDA's expectations and provide clarity and direction, which was lacking early on. There is an additional benefit within FDA, as guidance tends to enable a more consistent and comprehensive assessment. In the end, guidance development to achieve these goals is an essential piece of working collaboratively with stakeholders to address this true public health concern. So we'll go on to the evolution of FDA guidances. Hopefully you all can see this slide. We went on to publish our first nitrosamine guidance, control of nitrosamine impurities in human drugs, or what I'll refer to as nitrosamine guidance for the remainder of my presentation, in 2020, and subsequently updated that guidance in 2021 to update a deadline. At the time, our comprehensive understanding addressed small molecule nitrosamines and described root causes and provided API-focused recommendations or drug substance-focused recommendations. Though the guidance was destined to become outdated with the quickly expanding scope of understanding, it introduced the three-step mitigation strategy, which was adopted internationally and remains alive and well today. This high-level concept provided a sequential framework for mitigation that we continue to build on today. Step 1 recommends manufacturers perform a comprehensive risk assessment to evaluate potential nitrosamine impurities in the drug product. Records of this risk assessment should be maintained on site and available upon request. Step 2 recommends that if a risk or any risk at all is identified, that manufacturers perform testing to confirm the presence and levels of nitrosamines. Step 3 recommends that based on those testing results, manufacturers make and report changes to prevent or reduce excessive nitrosamines per applicable requirements. These recommendations apply to chemically synthesized APIs in application products, regardless of where it is in the application lifecycle, as well as OTC products. All right, in 2023, we published the second guidance, titled Recommended Acceptable Intake Limits for Nitrosamine Drug Substance-Related Impurities, or what I'll call RAIL, the RAIL guidance, moving forward. At this time, we were increasingly concerned of the risk that NDSRIs pose to the nation's drug supply. That concern persists today. Within this guidance, the RAIL guidance, we introduced a new concept to predict AI limits for NDSRIs based on their structural features using a model called the Carcinogenic Potency Categorization Approach, or CPCA. I mentioned earlier the challenges our toxicologists faced when deriving acceptable intake limits. So this predictive model, which was eloquently described in the RAIL guidance, is the basis for more than 300 potential or hypothetical nitrosamines that are listed on our Table 1 of the Cedar Nitrosamine Impurity AI Limit webpage. This achievement was welcomed by industry counterparts, as it provides clarity on what limits manufacturers should target. Finally, last year, we published the second revision of our 2020 nitrosamine guidance, which provides some key updates impacting NDSRIs that I'll address soon. But first, we've reached our first challenge question. All right, so FDA's recommended three-step mitigation strategy includes all of the following except: perform risk assessment, submission of risk assessment to FDA, perform confirmatory testing, report changes to FDA as appropriate. Hands for A? B? All right, I see many hands. C or D? 100%. Thank you all. The answer is B, of course. Remember, the three-step mitigation strategy recommends maintaining a record of your risk assessment at your manufacturing facility and available upon request, not necessarily submitting it to FDA. Now for some key updates of our nitrosamine guidance, updated last year. First key update simply broadens the scope of the nitrosamine guidance to include NDSRIs alongside small molecule nitrosamines. Manufacturers should assess the risk of NDSRI formation, which occurs primarily in drug product manufacturing, in addition to evaluating the risk of small molecule nitrosamines, which primarily form an API synthesis, following the same three-step mitigation strategy. Second, the second key update is related to nitrosamine controls, and you'll see it on the screen here. This update introduces a flexible approach for calculating AI limits when multiple nitrosamines may be present. Where we previously recommended that nitrosamines, when multiple are present, be controlled to the limit of the most potent nitrosamine, we are now suggesting that manufacturers instead ensure that the levels of nitrosamines, when totaled, should result in an exposure level that does not exceed the acceptable increased cancer risk of 1 in 100,000. And there's an equation on the screen there on the left side for you. Here, you'll see revised nitrosamine guidance updates control recommendations with a clarification on method sensitivity to align more closely with recommendations of ICH Q2R1. When our first version of the nitrosamine guidance recommended a level of quantitation of 0.03%, 0.03 ppm, we found that it would be more practical to revise the recommendation such that the level of detection and level of quantitation are commensurate to the level at which the impurities must be controlled. The third key update I'd like to bring to your attention today provides some recommendations for implementing changes based on confirmatory testing results. You will see, again, that our new recommendations continue to build on the three-step mitigation strategy of risk assessment, testing, and reporting changes. Now, let's assume that NDSRIs form. We recommend that you perform confirmatory testing. And depending on those findings, we recommend that you submit changes to your application as illustrated here. If confirmatory testing finds nitrosamines exceed the corresponding FDA recommended AI limit, we recommend that you make changes to reduce or mitigate the impurity and submit those changes to the agency for review as a prior approval supplement. If confirmatory testing finds nitrosamines exceed 10% of the corresponding FDA recommended AI limit, but does not exceed the FDA recommended AI limit itself, we recommend you implement routine testing and submit those changes to the application as a CBE 30. If confirmatory testing finds nitrosamines do not exceed 10% of their corresponding FDA AI limit, we find that routine testing may not be necessary and recommend you submit those findings as an annual report. Sharing these reporting recommendations, I don't mean to exclude that there are others that are still in place. So you'll see here that these include timely reporting of observations of excessive nitrosamine levels to FDA through field alert reports in accordance with 21 CFR 314.81, and timely reporting of discontinuances and interruptions in your manufacturing by contacting CDER's drug shortage staff in accordance with Section 506C of the FD&C Act, as this allows FDA to consider as soon as possible what actions, if any, we need to avoid shortages to protect the health of patients who depend on your products. So let's take a quick detour with the second challenge question. All right. Which of the following statements is not true? FDA recommends that applicants perform confirmatory testing if a risk assessment indicates an impurity is at a low risk of formation. FDA recommends that applicants routinely control nitrosamines if testing results indicate that nitrosamines are found at less than 10% of the corresponding applicable FDA recommended AI limit. FDA recommends that applicants routinely control a nitrosamine if testing results indicate that nitrosamine is found at greater than 10%, but less than equal to than the corresponding FDA recommended AI limit. And D, FDA recommends that if applicants find nitrosamine impurities in their product exceeding the recommended AI limits, they mitigate and report changes as a prior approval supplement. Alternatively, you may provide justification for a different limit via PAS. I'll give a second for folks to see these responses. And anybody for A? What about B? All right, I see hands for B, C, and D. All right, you're right. So pardon if this question is a little tricky, but remember, even low risk is considered risk. All right. So I'd like to briefly bring your attention to an essential resource that we developed to be your landing page for nitrosamine-related information. For comprehensive information, visit CEDAR's nitrosamine impurity acceptable intake webpage, which includes links to guidance documents, acceptable intake limits, interim market-wide regulatory discretions, which are only applicable to approved products in the market, and these interim limits are not supportive of a permanent control limit. We also include analytical procedures that are posted in a section entitled Emerging Scientific and Technical Issues, where we plan to periodically communicate new information, understandings, and update timelines. And also a very useful revision table that shows each time that the webpage was updated. You'll see a screenshot here of the front of that page, which also has an opportunity for you to subscribe to receive email notifications about updates, ensuring that you stay informed on the latest developments. So back to the updates. Our nitrosamine guidance was revised to recommend some of the following strategies to mitigate NDSRI formation in your drug product, as previous versions only were API focused. So the recommendations include screening for excipients, adding antioxidants, adding pH modifiers, and exploring innovative strategies. Our scientists are eager to evaluate those new approaches that you propose. As we learn more about the root causes of NDSRIs, it became apparent to our experts that the product reformulation would be a powerful tool for mitigation. Which brings me to the fourth key update on the nitrosamine guidance that emphasizes reformulation as a mitigation tool. So when you're reporting reformulations, we recommend that you submit data for at least 3 batches, accompanied with at least 3 months of stability data. If our analysis sees the potential for an upward trend for out of specification, we might request 6 months. And long-term data for the first 3 production batches should be reported through the proposed shelf life and annual report. So in addition to these CMC recommendations, the guidance includes recommendations for alternative BE approaches that may apply to your proposed reformulation application. So with reformulation on the table, we realized that in certain cases, this type of change necessitates in vivo bioequivalent studies. So FDA scientists went on to research the impact of adding small amounts of antioxidants on nitrosamine formation and permeability. And additionally, research funded by FDA through the CERC initiative has shown small amounts of antioxidants do not affect intestinal transporter activities. All these together, as long as it's in accordance with 21 CFR 32024, may provide alternative approaches to show bioequivalence without in vivo studies. And if you have more questions about that, you can reach out to an expert in bioequivalence or read our guidance for more details. So I have some future considerations for your thoughts. We don't know what's going to happen in the future, but these points may be important. As soon as I feel like we had a hold of the small molecule nitrosamines, NDSRIs popped up, so we want to know, we're not sure what's next. So continued advancements in analytical procedures and research on health impacts will enhance our understanding and support control measures. Industry partners have been doing a lot of this already, but it's important to consider these points to implement comprehensive risk assessments, especially for products at high risk of potential potent nitrosamine formation. Engage proactively with regulators and invest in research technology for effective nitrosamine control, because we're not yet out of the woods. Until then, we expect manufacturers to adopt the best practices and consider our guidance recommendations, continue to report issues as they arise and remain responsive to our inquiries, maintain your commitment to patient safety and quality control, and collaborate with us on nitrosamine control measures. So this morning, we covered the details that you see here in these 7 slides. And in closing, let's continue our commitment to patient safety and quality control by adopting best practices, staying informed through resources like the CEDAR Nitrosamine Impurity Acceptable Intake webpage, and collaborating to address nitrosamine challenges in the future. I extend my gratitude to the colleagues across the agency who support this work, with special thanks to Susan Zuck and Dr. Dongmei Liu for their assistance in preparing this presentation. Thank you also to the GDF committee, and thank you. If you have any questions, feel free to get in touch. Thank you. Oh, I'll pass it on to the next speaker, Dr. Kaur.
Good morning, everyone. Thank you, Corey, for the kind introduction. In today's presentation, I will talk about bioequivalence approaches for nitrosamine-impacted generic drug applications, along with the case studies. This is my disclaimer. To begin with, I will start with the background and bioequivalence approaches for drug products impacted by nitrosamine impurities, followed by case studies, and will wrap up with a summary. To begin with, since 2018, several commonly prescribed drug products have been found to contain levels of nitrosamine impurities at unacceptable limits, with some classified as probable human carcinogens. This nitrosamine issue has impacted a wide range of drug products across various dosage forms, including both new drug applications and abbreviated new drug applications, NDAs, being the majority. To mitigate the risk of nitrosamine impurities, the industry may need to take various approaches, but not limited to changes such as change in API, drug product manufacturing process, and change in the formulation by changing the amount or type of excipient. The FDA has developed an alternative BE approach specifically for reformulated drugs products of nitrosamine impacted applications. This approach is outlined in section called Bioequivalence Studies for Reformulated Products of the Guidance for Industry, Control of Nitrosamine Impurities in Human Drugs, finalized in September 2024. I will call this guidance as nitrosamine guidance. As Dr. Ray pointed out, the recommendation in this guidance is based on the research conducted by FDA in collaboration with external researchers, which indicated that the addition of certain antioxidants or pH modifiers to the formulations may significantly inhibit nitrosamine formation. Notably, the incorporation of small amount of 4 antioxidants, ascorbic acid, alpha-tocopherol, propyl granulate, or cysteine, had no significant effect on the permeability of BCS class 3 model drugs and did not affect intestinal transporter permeability. Activities. Please note that this alternative BE approach is not applicable to narrow therapeutic index drugs, sublingual, buccal, or orally disintegrating dosage forms. This table summarizes the bioequivalence data recommended to support formulation changes under various scenarios based on the recommendations in the nitrosamine guidance. In general, when the applicants reformulate by using one of the antioxidant studied in FDA research or a pH modifier at a level not more than 10 milligram per dose or maximum daily exposure, whichever is lower, it is recommended to submit comparative dissolution data on the pre and post change products in quality control, QC, dissolution medium, and multimedia of pH 1.2, 4.5, and 6.8. This applies to IR, solid oral, or oral suspension dosage forms containing BCS class 1, 2, and 3 drugs. For scenarios beyond these, additional in vitro bridging studies or in vivo studies may be needed. These cases will be evaluated on a case-by-case by the agency. Now we will go through 4 case studies. In the first case study, which is related to an approved ANDA for an immediate release capsule available in 3 strengths to control the risk of nitrosamine impurity, the applicant proposed change in the grade and amount of the filler, as well as removal of the glycant, which led to significant reduction in the fill weight of the drug product. To support these changes, the applicant proposed to submit dissolution data on the pre-change versus post-change test product in QC medium only. The agency determined that there is no addition of antioxidant or pH modifier. Therefore, the alternative bioequivalence approach recommended in the nitrosamine guidance is not applicable in this case. Per SUPAC IR guidance, removal of glycant constitutes a level 3 change. And per the product-specific guidance for this drug product, a FED BE study is recommended on the highest strength and the lower strengths are eligible for the waiver. Based on the principles in the SUPAC IR guidance and the recommendations in the PSG, the applicant was recommended to conduct FED BE study on the highest strength and dissolution data comparing the pre-change versus post-change test products in QC medium for all 3 strengths. This case study too is related to an immediate release tablet dosage form, which is an approved ANDA available in 2 strengths. In this case, to mitigate the risk of nitrosamine impurity, the applicant proposed to add an antioxidant and accordingly adjust the level of filler microcrystalline cellulose to maintain the constant tablet weight. Similar to case 1, in this case also the applicant proposed to submit only QC dissolution data for pre-change and post-change test products. Based on the information available to the agency regarding the solubility, a definitive determination could not be made whether the drug substance belongs to BCS class 3 or 4. Proposed antioxidant was one of the antioxidants studied in FDA research. The proposed amount was less than 10 milligram per day, and the proposed level was within the maximum daily intake levels in FDA-approved drug products for adults, but it exceeded the level for pediatrics for similar context of use. Based on the agency's evaluation, the applicant was asked to confirm that there are no other changes such as manufacturing site, process, etc. Submit dissolution data in QC and multimedia for each strength for pre- and post-change test products. Solubility data as per the recommendations in M9 BCS guidance to demonstrate high solubility of the drug substance used in the test product. If high solubility is not demonstrated, submission of additional data is required to demonstrate that the antioxidant at the proposed level in the post-change test product does not impact the absorption of drug substance as compared to pre-change test product. This data can be based on the results of the studies such as validated IVIVC, PBPK modeling, or any other studies as appropriate. Justification data to support the safety of the antioxidant at the proposed level in pediatric population. In case study 3, which is a delayed release capsule, it's also an approved ANDA. It's available in multiple strengths. The applicant proposed to add an antioxidant, a pH adjuster, and accordingly reduce the weight of sugar spheres to keep the approved capsule weight. In this particular case, in addition to submitting the dissolution data, the applicant proposed to conduct CACO2 study to prove that antioxidant is not expected to have an impact on API absorption. Agency determined that the drug substance belongs to BCS class 3. The proposed antioxidant is one of the antioxidants studied in FDA research. The proposed amount is acceptable. In terms of the formulation design, delayed release formulation consists of an IR drug layer and enteric coating layer. The antioxidant is added to immediate release layer only, and the change in amount of sugar spheres falls under level 1 change. There are no other changes to the composition or manufacturing process. Therefore, based on the totality of evidence, it was determined that the risk of change in drug absorption by antioxidant in the IR drug layer is very low. Therefore, submission of CACO2 permeability study is not needed. And the same was communicated to the applicant. In addition to providing the dissolution data in QC media, the applicant was recommended to provide dissolution data in multimedia of pH 1.2, 4.5, and 6.8. This last case study is related to approved ANDA for fixed dose combination drug product containing API A as IR formulation and API B as extended release. In this case, the applicant proposed multiple changes, mainly 4 changes: addition of an additional purification step for API A, addition of an antioxidant, change in processing solvent used during granulation, and amount of non-release controlling excipients used in the extended release part. And applicant asked agency about the type of data needed to support these changes. The agency determined that the proposed type of antioxidant and its amount are acceptable. Both the APIs belong to BCS class 3. The change in type of processing solvent as well as amount of non-release controlling excipients was considered a level 3 change. Therefore, in this particular case, the applicant was requested to conduct a single dose in vivo fasting BE study comparing the highest strength of the reformulated test product versus reference standard, submission of dissolution data for pre-change versus post-change test products for all the strengths in QC and multimedia. To summarize my presentation, to apply an alternative B approach in bridging the drug product changes, it is crucial to thoroughly evaluate the physiochemical properties of the drug substance, level of the changes, and formulation release mechanism. I'd like to thank Dr. Bing, Dr. Honglin, Dr. Rong, Office of Bioequivalence, Nitrosamine Working Group members, and the divisions of bioequivalence assessment teams who assess these inquiries. Thank you.
I think your questions, and I would like to hand over to Carl. Thank you.
Of course, thank you. I'd like to introduce our presenters up to the stage for the panel discussion. let them take their seats. While they sit down, just to remind everybody of a few reminders from yesterday. So if you don't mind, if you have in-house questions, go ahead and take a moment to line up at the 2 microphones. You can see them right here by these large pillars that are in the room, so they're easy to find. Go ahead and line up too if you have any questions that you might want to ask. As soon as you think of them, go ahead and line up. That way we can gauge between the in-house questions and the online questions that we'll have. But we'll go ahead and begin with these. And again, if we have any questions, we just ask that you keep these relevant to today's presentations on this. If we have anything that we do need to defer, we'll make sure to get you an email contact address as well for any follow-ups. So at this time, we'll begin with the in-house questions. We have one right over here.
Hi, thank you. Very nice presentations. I know regarding the guidances related to the nitrosamine, we all know, as you said, many of these guidances were updated, but took a lot of time. So some of the things I would be proposing is to do proactively. One is, maybe you can take this message to the team that we are still not clear about the pathway of what happens if we find something that is higher than the CPCA in the rail guidance and have a justification. Because what is happening is they say, go to the drug shortage. Drug shortage is radio silent about it. Like there are drugs where for 1 year people wouldn't hear anything. So now they don't know what to do. They're trying to find their ways. So to find, to give some alternatives, because FDA in the guidance does say that CPCA is not the last word. You can try other things. However, the pathway with that other things are not clear and very difficult, and people are struggling with that. And we are worried that there are certain drugs that will disappear from the face of the earth, if these are not addressed, some very good antibiotics. The other thing is FDA, took a long time to eventually agreed to the LOD, LOQ, which was much more scientific with EMA. In 2020, the guidance, basically they gave a random number based on valsartan, but then EMA already had that calculation, that they have now adopted. So other thing is EMA, Health Canada, and Visa, they have de-risked several nitrosamines based on, the transgenic mice assays and other studies. And it may be a good idea also to take that message that we do need collaboration and, uniform address. It can't be that, people are dying somewhere.
For the sake of time, we do only have 2 minutes.
This is very important,
though. Is there a question that's associated with this? Yes.
No, I think let me finish because people need to understand. I'm very sorry. Just let me. Please talk to them that they should try to collaborate with other teams. I'm sorry about this, but this is extremely critical.
And we'll take that note, but for the sake of time, we'll go on and move on to another question.
Thank you. I have a question regarding the nitrosamine. We know that from FDA, we have a NDASLRI database routinely updates, and also the guidance expect the industry to monitor this database. So my question is that, is there any specific reaction timeline for the industry after the NDASLRI limits published into the database?
Thank you. That's a good question. So just so I can make sure I understand. So with revisions to the guidance, new nitrosamines are added, and you'd like to understand if there's some timeline kind of flexibilities for those to be addressed. Is that correct?
Yes.
Okay.
Because we know from the FDA website there is a database for NDASLRI limits. Yeah.
Absolutely. So at this time, we have a August 1, 2025 deadline for manufacturers to address the issue, which would be if you find it in a risk assessment. And typically, if we post something on our webpage that assumes that there is a risk, you would perform some confirmatory testing. I'm not aware of any flexibilities beyond that date at this time. However, if we do have it, again, that emerging scientific and technical issue section on our webpage will be updated to reflect those revisions.
And may I know what if the NDASLRI limits published after August 2025?
It's a good question. I'm expecting that we'll have some considerations for that. we would expect that firms have some time to address the issue. We know that method development can take months, if not years. And so we would actually make sure that we're flexible with manufacturers if we're to add something after the deadline.
Okay, thank you.
Thank you.
Excellent. We'll have one more in-house question, then we'll switch to online. So if you'd like to go.
Hi, I just have a follow-up question of that. So if a drug product manufacturer is unable to complete the confirmatory testing or implement necessary changes by August 1st, 2025, how can a manufacturer proceed with the ongoing operations?
That's a good question. I would recommend that you contact the agency for these issues. Perhaps general correspondence to your application informing us that you need some more time. That will be beneficial for us, and we're happy to work with manufacturers on this because we want to maintain drug supply of these critical medications.
Okay, thank you.
Excellent. Thank you. And we'll now take a question from online.
Yes, thank you. This question is for Dr. Kuna. For comparative assessments between a generic drug and the RLD, is it mandatory that the RLD be sourced from the US market? Or is it acceptable to use the same RLD product purchased in another country, provided that it is manufactured in the US and marketed under the same brand name?
Thank you for the question. So if I understand, so the question is about can the applicant use the RLD from a different market?
That's correct.
So the agency recommends that the RLD is used from the US market because we don't know, the RLD that was approved in other market. So we don't have any information on the quality of that.
The question specifically says that the RLD be sourced from the US market. And is it acceptable to use the same RLD product purchased in another country, provided that it's manufactured in the US and marketed under the same brand name? I don't know if that puts a little bit of a tweak on the question.
If it is marketed in another country, but it's sourced from the US?
Vice versa. Let me say it again. For comparative assessments between a generic drug and the RLD, is it mandatory that the RLD itself be sourced from the US market? Or is it acceptable to use the same RLD product if purchased in another country, provided that it is manufactured in the US and marketed under the same brand name?
I believe it is acceptable to use from the other market.
Hold on 1 second. So
Yeah.
based on US regulations, it is, oh, you want me to go on?
Yeah.
No worries. Just stepping in shortly, based on US regulations, it is requirement to utilize a reference listed product that is registered by the FDA in order to establish that bioequivalence. So it is a requirement. It's not about purchasing, it's about being approved and registered through the FDA. Thank you.
And would you mind introducing yourself, please?
Sorry?
Would you mind introducing yourself? Introducing yourself.
Sarah Ibrahim, Associate Director for Stakeholder and Global Engagement, Office of Generic Drugs. Thank you.
Thank you, Dr. Ibrahim. Excellent. Thank you for that response. We'll take the next in-house question, please.
Thank you. Good morning. This is for Dr. Ray. So I work for a Acuon lab, contract testing lab. So till now we did over 250 method validations or verifications for both the nitrosamines and NDSRIs. Still, sometimes we do face challenge in achieving 0.03 ppm LOQ or 10% of the specification if it's at a high dose, higher dosage formulation or a category 1 or combination of it. So I would like to seek advice from what is advisable to acceptable to achieve that LOQ, and if we have to include it for routine testing.
Thank you. I'm not a method evaluation expert. We have our colleagues in the Office of Pharmaceutical Quality Research, who I hope I can get a response to you with more detail on this. But I'm aware that there are some methods that you can use to concentrate certain material And there's ways that you can maintain some level of sensitivity that way. But again, this is not my level of expertise, and I do appreciate that there are challenges that remain when it comes to method sensitivity.
Sure, thank you.
Excellent, thank you. And our next in-house question, please.
Hi, good morning. Yeah, my question is regarding the reformulated product to mitigate the NDSRIs submitted as a PAS. So the 0, 1, 2, 3 stability data to be submitted, can it be on the R&D scale batch? Because now we are in the crunch of a time. Is that expected on the registration batches?
I believe you can do, scale batch or registration batches for 3 months?
Yeah, because we are doing the R&D experimentation first to see the formulation is working out or not. By the time we are confident with the data, if we want to again execute in the plant and wait for the 3 months data, the time is passing, and the August 1st is the date for the compliance. So.
That's a good question. I'd say that we would want to see commercial batches, but I'm not 100% sure. So I'll talk with you offline if that's okay, and then we can get some answers for you.
Yeah, sure. Thank you.
Can I get your name?
We'll collect that. Yes. Okay. Excellent. We'll go to our next online question this time.
Yeah. My name is Raghu Raman. I'm from Sygen Pharmaceuticals. And yes, and only one small follow-up question. So August 1st compliance is for the batches manufactured after August 1st. Because you have interim limits for so many products.
I'd say if you manufacture before August 1st, that still works.
No, I'm saying suppose if you have fluoxetine, diltiazem, you have some interim limits. Mm-hmm. And you have a different limit after August 1st. So what are the batches manufactured before August 1st still
for the sake of time, can we go ahead and switch to our next online question? We'll handle this, we'll discuss it offline. Thank you.
Very interactive session here. All right, this question is for Dr. Kantev. What level of method validation is required for nitrosamine confirmatory testing? It's a two-parter. And can methods be suitable for use at that point and only fully validated if the routine testing is required? Thank you.
Thank you for that question. I'd say that method validation, full method validation would be required for routine testing. You can submit that to the agency for review.
Excellent, thank you. We have our next in-house question here.
All right. I just have a question in regards to general NDSRI limits. So in a database, there are quite a few unstable NDSRIs where reference standards are unavailable, and sometimes we can't even develop a method because they're so unstable. So is there any agency directive coming up or anything to help the industry with these unstable impurities?
Yes, thank you for that question. So the nitrosamines that are listed on our webpage are listed as theoretical nitrosamines. So we do understand that concern that they may not necessarily be forming, and these are volatile reactions. So we would accept justification that it's not forming through forced degradation reactions, or you can provide justification that you're unable to purchase a reference standard. That information is very helpful to us, and may be helpful to communicate to other manufacturers as well, because we are working on potentially removing some of these theoretical nitrosamines from the webpage once we have sufficient evidence that they do not form.
All right. Thank you so much.
Excellent. We'll take our next online question, please.
This question is for Dr. Kaur. What is the form of inquiry for the industry inquiries and the BE proposals shared in the case studies? For example, controlled correspondence, product development meeting, etc.
I think you'll need to make sure the light's red.
Sorry. All these case studies that I presented today were from controlled correspondences.
Thank you.
Excellent. And I think we can take one more online question since seeing some familiar faces here, and then we'll go back to in-house.
Okay, let's go to Dr. Kantev. Let's see. The FDA recommendation for 3 batches with stability for reformulation is interpreted to be applicable to major reformulation changes. Would you confirm that changes to excipients are considered minor and could be submitted in an annual report?
question is for?
It's addressed to Dr. Kantave. The FDA recommendation for 3 batches with stability for reformulation is interpreted to be applicable to major reformulation changes. Would you confirm that the changes to excipients are considered minor and could be submitted in an annual report? Anyone is free to answer if it's applicable.
Okay, I can take that question.
Okay, thank you.
Not all the time the changes to excipients are considered minor, so you may refer to the spec guidances. And depending on the level of the change, the requirements of the data are different. Thank you.
Thank you.
Excellent, thank you. And we'll go to our next in-house question.
I have one more question to Dr. Kantave. For the extractable and leachable nitrosamine impurity from the packaging materials, for the oral solid dosage form, can we consider it as a low risk, or we should have further evaluations?
I think I'll defer on that question.
Okay. Excellent, thank you. We'll go to our next in-house question.
Hi, Dr. Ray. So could FDA clarify the scientific and the regulatory rationale underlying the derivation of the interim AI limits?
For scientific rationale for derivation of interim AI limits?
Yeah, interim AI limit.
Yeah, I think I'll also defer on that one.
All right, and at that time, we'll go ahead. I think we have time for one more online question if we'd like to take one.
Lots of questions coming in. Let's do Dr. Kaur. What's the FDA's definition of technical grade? For example, if an excipient source change does not impact the approved specification tested for USB with differences in particle size distribution, which is not an approved specifications criterion and not a CMA, is this considered same grade? Dr. Kaur, would you like me to repeat?
That question is for me. Changes in the grade, right? In terms of specifications. So probably I will defer to CMC reviewer, but say for example, you have hypermallows, which comes into various different grades, which may not be considered same. So you have to really look at, they may differ in the viscosity, molecular weight. So can I say the one which has very low viscosity versus one which has very high viscosity is going to behave similarly? Probably not.
Thank you much.
All right, and it looks like that's all the time that we have questions for today. And so if you did have any additional questions that weren't addressed, we'll make sure to give you a contact email address after this that we could have those for any questions that are additional beyond this. But at this time, I believe there should be a break coming up soon. Let me see. Yeah, so we'll take a quick break. I think it's about 20 minutes here, right? Is the typically. So at that time, we can come back at, let's see, that would be 10:15, I believe. And so, perfect, 10:15, we'll see you back.
Right, if everybody could take their seats. Okay, if everyone could take a moment to take their seats, return back to their seats now. All right. Okay.
This is where you sing and dance.
Let's say, I hear the bells. Yeah, perfect. Okay. All right, now that it looks like mostly everybody's back in the room taking their seats, I'll go ahead and begin our next session. Thank you so much for returning back. So next, we have our next 4 presenters coming in. The first will be Dr. Diana Vivian, Associate Director of the Division of Bioequivalence 2 within the Office of Bioequivalence, within the Office of Generic Drugs within CDER. Our next speaker that we'll hear from is Dr. Darby Kozak, Deputy Director of the Office of Generic Drugs within CDER. Next, we will hear from Dr. Jaeyoon Kim, Senior Pharmacologist within the Division of Bioequivalence 3, within the Office of Bioequivalence, within the Office of Generic Drugs at CDER. And last, we will hear in this next session from Dr. Ying Fan, Team Lead in the Division of Clinical Review, or DCR, within the Office of Safety and Clinical Evaluation, within the Office of Generic Drugs within CDER. So at this time, if you will help me and welcome Dr. Diana Vivian, I'll let her take the stage.
Thank you for that nice introduction. I'm going to be speaking today on one aspect of the impact of ICH M13A implementation on bioequivalence assessment, and specifically regarding the removal of data due to low exposure. This is a standard disclaimer that this presentation reflects my own views and should not be construed to represent FDA's views or policies. The learning objectives for this talk are 1, to provide an overview of the ICH M13A guidance recommendations regarding the removal of BE study data due to low exposure, and 2, to highlight some case studies showing how OB has implemented these recommendations, including how the scientific principles have been applied for non-immediate release products. So what was our previous practice regarding low exposure data removal? We have 2 FDA guidances that address this. The draft guidances for industry, BE studies with pharmacokinetic endpoints for drugs submitted under an ANDA, which was published in August 2021, and also statistical approaches to establishing bioequivalence, published in December 2022. Data should not be removed from BE studies due to identification of statistical only outliers. And such data was generally only removed along with real-time documentation of a protocol deviation. Otherwise, it could be unclear if the anomalous data resulted from other factors such as product failure. However, in limited cases, an exception was made due to if all of the subject data for one period was below the limit of quantitation and we could not calculate the PK parameters for that period, it disallowed for the use of that subject in statistical analysis, which is a little bit similar to what we're seeing in ICH M13 with some additional caveats. But in that case, it was not possible to calculate PK parameters for the subject without something like imputation, which comes with its own limitations. And it also is likely that in these cases, there was a compliance issue and it's not actually due to the product. So you can see here the M13A guidance, bioequivalence for immediate release solid oral dosage forms, was published in October 2024. And I'm going to be speaking mainly about the removal of BE study data due to low exposure, which can be found in section 2B under data analysis for non-replicate study design. This guidance says that generally data should not be removed from the statistical analysis of BE studies solely based on statistical tests that identify extreme values in the PK variables, which is consistent with what you're seeing in the other guidances. There is also language about an exception that can be made for a subject without measurable concentrations or only very low concentrations. And this is the new part in this guidance. Some more detail on this. It could be applicable for either the test or the reference standard product. Specifically, it sets a parameter for AUC for that period. So this could be applicable when AUC for a period is less than 5% of the geometric mean AUC for the drug product overall. And the geometric mean AUC should be calculated without including data from the low exposure subject. Because this is considered to be the result of subject noncompliance, exclusion will only be accepted in exceptional cases, in general, with no more than one subject in each study. And of course, applicants should proactively minimize the potential for subject noncompliance in the conduct of the BE study through their study design and protocol, for example, by mouth checks after dosing to make sure that this is minimized as much as possible. And even if an applicant removes subject data because of low exposure, the data should still be submitted for agency assessment. Now I'm going to get into some case studies of how we've implemented this in OB. The first case is for an immediate release product with a two-way crossover BE study. And you can see the individual subject data profile shown here, where period 2 of the reference product is much lower than this subject's period 1 data of the test product. So after reference standard treatment for this subject, the observed AUCT was 3.58% of the geometric mean AUCT for the reference standard overall. And like I mentioned, this is excluding the low exposure subject for that calculation. When including the subject, both Cmax and AUCT failed to meet BE criteria. You can see at the higher end of the confidence interval there, they fail. When excluding this subject, it brings the confidence interval dramatically differently into range. For this case, the removal of the low exposure data was considered acceptable because it met the scientific principles outlined in ICH M13A, including that the AUCT was less than 5% of the geometric mean AUCT for that product. And when the low exposure data is included, you could see that the TR ratio for AUCT, AUCI, and Cmax were close to 1, and the 90% confidence intervals were all within BE limits. And further, based on available data, such low concentration is not considered to be typical of the reference standard. Case 2 was for an IR product with a fully replicated BE study design. In this case, a reference scaled BE approach was adopted due to high variability. In the individual subject data, here you can see the period that the applicant wanted to exclude was that shown in red, the lowest reference product treatment. It's not as obvious as the previous case where it's actually pretty close to the period 1 reference product data also, and we'll get into that. The AUCT of that period compared with the geometric mean AUCT overall was 7.6%. And the AUCT and Cmax results failed to meet acceptance criteria using the reference scaled approach when this subject was included, but it met if excluding the subject. However, in this case, it was not acceptable. For one, because the AUCT was greater than 5% of the geometric AUCT of this product in the study. And also due to the highly variable nature of the drug product, similarly low concentrations were observed both within this ANDA and across other ANDAs, and were not excluded in other cases from statistical analysis. And it really suggests that the observed low exposure is not likely due to subject noncompliance. So this addition in ICH M13A is meant to account for really realistic subject noncompliance that we've seen over the years, but it's not meant to just be a statistical outlier for something that may be due to product performance. And the applicant in this case was recommended to repeat the study. Case 3, although ICH M13A is not for modified release products, we'll show how it was used in this case for, using the scientific principles. This is a modified release product with a two-way crossover BE study. I think you can see in this case, I think there were only 5 measurable concentrations for period 2 of this subject, which was given the test product, compared with a more typical profile for the reference product. The AUCT was 1.57% of the geometric mean AUCT for the test product, and including this subject, AUCT, AUCI, and Cmax failed to meet BE criteria on the lower end. So although M13A is not applicable generally for MR products, we used a risk-based approach using the scientific principles outlined in the guidance and some other risk considerations you can see here to evaluate this. For one, the AUCT in this case was less than 5% of the geometric mean AUCT. The concentrations we observed were very close to the LOQ. Such low concentration was not expected based on the overall variability of the product. Excluding the subject, the TR ratios for AUCT, AUCI, and Cmax are very close to 1. And we looked at some other risk of the product overall, including that there was a similar formulation between the test and reference standard products with the same type and grade of release controlling excipients. And there were comparable dissolution profiles between the test and reference standard product in QC media and also multi-pH media. And further, this product is meant to be taken once daily with an evening meal, suggesting the already acceptable Fed BE study has more relevance potentially than this study. So this was all taken together as a totality of evidence approach to conclude that this was likely due to subject noncompliance and could be excluded. And the last case is for a narrow therapeutic index drug product, MR drug product with a fully replicated BE study design. In this case, the replicated BE study design was due to the NTI nature of the drug and not due to high variability. So for an NTI drug, the acceptance criteria include 95% upper confidence bound from reference scaled BE less or equal to 0, unscaled 90% CI within 80 to 125%. An upper limit of 90% equal tails less than or equal to 2.5. And you can see the profile here. The AUCT compared with the geometric mean AUCT overall was extremely low, only 0.27% for this one period, period 4 after test treatment. You're only seeing 2 periods here, even though it's a replicated BE study design because the subject withdrew from the remaining 2 periods. So we only had 2 periods of data to work with for this subject. Including the period 4 PK data, the AUCT, AUCI, and Cmax failed to meet acceptance criteria from unscaled BE assessment, but still met the reference scaled BE criteria. And again, similar to case 3, we used a totality of evidence risk-based approach to evaluate whether this it was likely due to subject noncompliance or could be something to do with the product. In this case, the AUCT was less than 5% of the geometric mean AUCT of that product. The 90% CI, when excluding the subject, were in a very tight range of 96 to 102%. I know we don't have an official narrow range, but it does seem very close to 100. And the observed plasma concentrations were all very close to the LLOQ of 0.05 microgram per ml. Further, we looked at the formulation and release mechanism of the test product, and it was very similar to the reference standard product with the same type and similar grade of release controlling excipients. Now I'll get into challenge questions. I think this one should be an easy one. Which FDA guidance document contains information about an exception for removal of BE study data due to low exposure? A, BE studies with PK endpoints for drugs submitted under an ANDA, published in August 2021. B, statistical approaches to establishing bioequivalence. C, M13A bioequivalence for immediate release solid oral dosage forms. Or D, M10 bioanalytical method validation and study sample analysis. I hope if you saw the title, you know the answer is C. And for challenge question 2, which of the following statements is not true? A, low exposure data removal may be applicable if AUC for that period is less than 5% of the geometric mean AUC for the drug product without inclusion of the data from the low exposure subject. B, data removal due to subject noncompliance may be applicable for the reference standard product only. C, exclusion will only be accepted in exceptional cases, in general with no more than 1 subject in each study. And D, all data, including any data excluded from statistical analysis for low exposure, should be submitted for assessment. And as we saw in the case studies, data removal due to subject noncompliance could be applicable for either reference standard or test product. So it's not limited to the reference only. In summary, the M13A guidance, bioequivalence for immediate release solid oral dosage forms, introduced scientific principles for the exclusion of low exposure BE study data, which is considered to be due to subject noncompliance. Removal of low exposure data will only be accepted in exceptional cases For example, if more than one subject in a study shows low exposure, this could bring other questions of dose administration reliability or product failure. And although M13A applies for IR products, the scientific principles of low exposure data could be considered for other types of products, as you saw in the two case studies that I mentioned, using a risk-based approach and totality of evidence to assess whether the low exposure data is in fact likely due to subject noncompliance or if there are other questions it raises. And I'd like to acknowledge the four folks here, especially Ming and Lucy, who helped me with a lot of the data analysis. Yep. And I'd like to welcome Dr. Darby Kozak.
Thanks, Diana. So as a reminder, we're always looking for ways to improve efficiency of our review processes, as well as the assessment sort of cycles to reduce the number of cycles before ANDA approval. As part of that, what we're looking for is gaps and current challenges to improve the ANDA submission quality. My talk, and which I hear was from a number of talks later this afternoon, is really to help communicate where we're seeing common ANDA major deficiencies, so that way industry is more aware of these sort of aspects, and as well as where we need additional communication and enhanced communication, and as well inform you where those aspects can be, so we can get higher quality ANDA submissions, improve our efficiency in terms of ANDA assessment, and get more first and second cycle approvals. So I've broken my talk up into three main components. The first one is I'm going to give a sort of retrospective look at where we are with ANDAs that were submitted in fiscal year 2018 and 2023, so that way you get what aspects we are looking at in terms of those approval rates, what we have as major deficiencies, and then do a little bit more of a tranching analysis. This can be done because there's a lot of data there, but I'll give you a few examples in particular of like what we look like for major deficiencies based upon generic industry size, as well as the complexity of drug product. The next thing I'll go into as second part is to then dig in a little bit deeper and look specifically at major BE deficiencies, where we're seeing different aspects there that you guys can work on, we can provide additional clarity on with respect to the BE. As I mentioned, other talks this afternoon, we'll go into DMF, CMC, and other BE aspects. There's a lot of rich data here. And finally, I'm going to finish up by talking about a lot of these initiatives we've been talking about the last few days, really to enhance that communication, that clarity, and get to that sort of next step where we want to be. So I know there's a lot of information on this slide, but like I said, there's a lot of information available to us. The more we know, the more you guys will be able to utilize and leverage this. What I'm looking at here is ANDA submissions in fiscal year 2018 and 2022, and the number of cycles that were done until approval, and as well as the current status. You might ask why we look at such things as so old as FY18. The reason there is we've now got 7 years of data. It's a more robust data set, so we can show you not only the first, second cycle, because all of them should have gone past that. We're going to look at third, fourth, fifth, and as well as the number of applications that are withdrawn. Then at FY22, we should have all those applications who have at least gone through the second cycle, so we can really look at that first and second cycle approval rates and better understand that. I think what we want to drive to, obviously, is first cycle approval, but there's still, we can see a lot of diversity within generic industry and areas we can improve communication. So I think right now, as a striving sort of desired goal, is we should always get towards second cycle approval. This means after your first CRL, that CRL should be clear. You should be able to address that CRL and get back to this next cycle and approval. Right now, we're getting at approximately 40% of ANDAs at second cycle approval. What we were looking for is you have this extra 60% of ANDAs that have to go either multiple cycles or get withdrawn in areas that we can potentially work on. The other thing I wanted to highlight here is the number of complex products we're getting per year. In FY18, we got about 14% of those ANDAs submitted for complex products. That's increased to 17% in FY22. And if I remember right, from FY24, we're up to over 20%. So we're seeing is the number of complex products being submitted to the agency is increasing and being proportional. So this is a good sort of aspect for those products in demand. So one thing that's always kind of asked is, well, if my generic industry has a lot of experience, theoretically our deficiency should be different. We have a lot of understanding. And what we wanted to look at then is, what are the relative major deficiencies with respect to your industry size? A very large industry we've classified as an industry that has more than 100 ANDA approvals. We then have large companies, medium companies, and small companies typically have 1 or 2 ANDAs already approved and or not approved yet, but no more than 6 ANDAs. And if you look over here on the right-hand side from those pie graphs, what we really see is in terms of distribution of major deficiencies, there's not a really big difference between industries. So any types of information that we can develop should be broadly applicable to all of generic industry, no matter if you're a large industry or company or a very small company. Also, one of the things that really pops out, and I'll talk about through a couple other slides, is the major sort of issue we constantly see as major deficiency is in the manufacturing. That includes your site facilities. Site facilities remains one of the major deficiencies that we see for applications and something that we definitely need industry to work more proactively on. Other aspects there is within the quality, drug product, drug substance, and also then bioequivalence. Interesting here, I don't know if you know a statistical aspect of that, but we also see that for complex products, in general, we're seeing a lot of very large industry going after that and very small companies going after these complex products. So the other aspect we want to look at is, well, what's a persistent deficiency? Do you mean what I showed you in the last slide is after the first cycle, what's the big sort of deficiencies we see? What is the thing that after a second, third, fourth cycle that it's still persistent as a major deficiency? And I think we'll show on this bar graph here, as you can see on the left-hand side, complete response 1, it's that general sort of distribution, BE, drug product, deficiencies overall, manufacturing. But as you go to second, third, fourth cycle, we're seeing is really improvement and a decrease in deficiencies in that BE, drug substance, drug product. But manufacturing and site facilities continue to be a persistent sort of issue through multiple cycles. I think this goes back to understanding why that's there and being more proactive in addressing those earlier and making it more appropriate. The other thing I want to kind of respond to is, well, complex products can obviously be challenging. So what do we see in terms of deficiencies in that sort of space? And we will say is that, your first cycle, complex products typically have, because we have more complex BE and product sort of things, they do make a little bit smaller percentage of those major first deficiencies. However, the manufacturing is regardless. It doesn't matter if it's a complex product or non-complex product. We don't really see a difference in terms of the percentage of manufacturing major deficiencies, depending on the product complexity. So now I'm going to do like a bit more of a dive, because I've shown you the overall the manufacturing. We hear from other groups talking about some of the quality issues and even other BE issues. But now what we're looking is just specifically at what are the major bioequivalence deficiencies that we see in that first cycle. And as you know, is what we're seeing a lot more is, and a lot more recommendations are these in vitro BE approaches. These are new approaches, characterization approaches. And so what we're seeing is in that area, there's still potentially a lot of unclarity or areas that we can provide a bit more information. And it's a sort of a growing area because the major BE deficiencies we're generally seeing are in these new in vitro BE studies, in particular, the in vitro release testing, in vitro permeation testing, and in vitro binding studies that are done. However, within that sort of scope, I do want to highlight the fact it's not necessarily the study design or everything else like that. It's what it is the method development, the discriminatory ability, all the additional information for the analytical components that go to help support the adequacy of those sort of studies that we're seeing that sort of aspect. The other second major sort of component we're seeing in major BE deficiencies is actually an insufficient validation data. This is typically with either those analytics for the PK or the in vitro sort of studies, in particular about sample matrix, stability, LLOQ, and chromatograph issues. And so this is another, in your method validation and analyticals, another key component that we see a lot of sort of gaps and challenges that can be addressed. So as I mentioned before, I keep kind of building this and going narrower and narrower and narrower. If we looked at BE deficiencies, you can think of any types of route administration, dosage form, or like that we can do a deeper analysis on. What I've chosen here is I've done now a deeper analysis of the actual in vitro BE deficiencies that are major for a specific route of administration, topical dermatological products. The reason we chose this is because more of these products are being recommended as a characterization-based or in vitro-based BE approaches. And we've seen of ANDAs submitted from FY 2018 to 2023, the number of ANDAs submitted proposing and or doing these types of in vitro studies has been increasing. What we're also seeing is a lot of deficiencies with those in vitro characterization studies. So this aspect here looks at not only those characterization studies, but out of all the characterization studies or in vitro studies, what are the ones that are still having the most major issue and or the issues with major deficiencies? If you go down to the bottom sort of pie graphs or donut graphs down below, it shows you both the in vitro release testing and in vitro permeation testing. So for those applicants in the dermatological space that are doing an in vitro release testing, we're seeing is some, probably about 33% or getting an adequate within that sort of first cycle, but there's still a large percentage that are inadequate. But then if you go to the in vitro permeation testing, this one that's done through a skin or sort of other sort of aspect there that's not synthetic membrane like the IVRT, we're almost exclusively still in an inadequate state. There's a lot of gaps still there to be able to understand and to be able to develop and to get that out so we can get to more first cycle approvals or second cycle approvals with those applicants. So this kind of pulls me into that third point. Now that we have this understanding of where these knowledge gaps are, where these challenges remain, where we need enhanced communication, I really want to advocate again, we've got a lot of these sort of aspects that we can start to have these engagements. And industry should be able to use these aspects, take it proactively, understand what your thing is, using these sort of resources, then to engage with FDA and or doing the research and background to make sure we're getting a high quality ANDA submission at that first cycle. So as the first and topmost, the GDUFA commitments provide you guys a lot of opportunities and resources to engage specifically about your ANDA application, both of the development stages. I think I'm probably preaching to the choir here, but as you guys know, and we've heard multiple times, control correspondence, product development meetings, and also other sort of aspects you can engage through us to get that sort of feedback about your application. Once you submit your application, you're going to get your DRLs, you're going to get information requests. This is going to give you better information right now, what you need to do in this cycle to get to there. If it goes to a complete response letter, you still have the opportunity to have a complete post-CRL meeting to get additional clarity. If you need the clarity, use these opportunities to get that at those multiple stages, increase that sort of submissions quality. The other one is what we're doing here today. It's using the opportunities of these workshops, of these sort of other sort of opportunities to engage with FDA and learn. All these are recorded, they're online. As you heard from SBIA, they're collating those. We've also created a website that collates these into an aspect, so you can then go back and learn from all these previous sort of aspects, ensure that we're addressing all those issues. So in addition to the SBIA, we also have a Center for Research on Complex Generics. They go in more detail, I think, about specific scientific components, engaging both academia and generic industry. All this sort of information is preserved on the websites. They can easily be accessed and tailored. And the last one is obviously our product-specific guidance program and our general guidance program. These are really just to be tools for you to be able to understand what the expectations and our current thinking are, and then to engage. Those are just recommendations. If you have other alternatives or proposals or different sort of thinking, use those communications such as CCs, control correspondence, or product development meetings to engage with us and talk about those alternatives. So this slide, again, has a lot of information, but what I want to do is give it that sort of snapshot. This snapshot is an example of the historic sort of information of enhanced communication and publicly available information just specific to those two studies that we mentioned for the topical dermatology products, the in vitro release testing and the IVPD studies. We understand that as we started to recommend these characterization studies, they may be new to industry, and therefore there may be this aspect where we need that sort of enhanced communication, understanding, and sort of working back and forth about what we need clarity for. So in addition to those draft guidances that were specific about IVRT for dermatological products that were released in October 2022 and the in vitro permeation testing, you also see that there was a whole series of SBI presentations and webinars even before we started recommending these things, preparing industry with this background information. This included the very original in 2020, identifying what are challenges and promises of these IVRT and the IVPT studies. Following up to that, you see 2021, we're already recommending and having seminars and workshops on what are best practices. And then looking at like, okay, now we've got these guidance out there. How do you interpret these guidances? Is there questions on these guidances in 2022 and 2023? In addition to that, as I mentioned, the CRCG workshops are usually 2-day or more intensive sort of things. We can actually do things like a step-by-step illustration, more hands-on. So really trying to identify what these gaps are, not only that, but how you guys can potentially address those issues. So on those websites that we've mentioned, as at the end of the sort of tables about this new helpful guidance and resources. This all hopefully links back to these and gives you a bit more sort of depth of understanding. So in conclusion, I want to really highlight the fact that, approximately 40% of our ANDAs that are submitted get approved by the second cycle. The rating 60% of the ANDAs, I think that's an aspect where industry and FDA need to identify what is it needs to be done more clarity, as well as industry needs to take more on to ensure that you guys are submitting high quality submissions, that when you get your first CRL, those deficiencies are clear and that you fully address them. So that way we can improve that efficiency. Each additional cycle is additional resources, not only on industry's part, but it's resources that FDA needs to use to do that sort of thing. So I think everybody's interest is to reduce the number of cycles and get those products out to the American public in as timely as possible. I'll remind you that the big one, most persistent, manufacturing facility major deficiencies. We really rely on industry to make sure that your facilities are accurate and up to date and adequate. With respect to the other sort of things, identifying those gaps within the BE drug product and bioequivalence where additional clarity may be needed, and or questions, using those GDF avenues to enhance communication to make sure that you guys have that sort of information that you feel is needed. And last, I will remind us in terms of for the GDF 3 initiatives, we really have those enhanced communication applications and encourage you guys all to use those. If you find them beneficial, let us know, provide feedback to us through multiple avenues to know what are your priorities, what are the things that you guys need additional information and clarity on, so that way you can improve your submission quality. So I will also advocate for that, the GDF Science and Research Program. There's a lot of the proactive information in this, allows us to get ready for those applications come review. You can see what our research priorities are here, and as well as helps prepare our review divisions. So I will come back in a little bit for the question and answer period, but with this, I'll hand over to Dr. Kim.
Thank you, Darby. Good morning, everyone. As Darby mentioned, my name is Ju-hyun Kim from Division of Bioequivalence 3. Today, Today, I'd like to talk about common BE deficiency in ANDA for ER drug product. Before going into detail of my presentation, I have a short disclaimer in the bottom of this slide. For next 15-minute presentation, I will start with the common BE deficiency for ER drug product, followed by first cycle BE adequacy rate and review time. And then I will share one case study with my recommendation. For learning objectives for today, I'm going to summarize common first cycle BE deficiency in ANDA for ER drug product. And then we are going to learn from the case study on BE deficiency in ER formulation to better understand the common BE deficiency impacting first cycle approval for ER product. And then we are going to identify a strategy to prevent some of this deficiency. Due to limit of time and resources, it was not feasible for me to look at ANDA for all ER drug product. So I focus on the tablet formulation, and then I selected the 8 drug product, which listed in the bottom of this slide. And this for 8 drug product, 157 ANDA was received until end of year 2024, and 224 deficiency was identified during first cycle BE review. This 224 first cycle deficiency can be categorized for different area, as you can see in the this slide. And most commonly observed deficiency topic was dissolution, and followed by bioanalysis and PK and formulation. So in each category, multiple deficiency was identified. I'm listing common deficiency in order of frequency and omitted some less frequent one. 37 deficiency was related to the dissolution. common issue was missing or inadequate multi-pH media dissolution and dissolution data for score tablet. For QC dissolution, usually no problem in submitting data, but the lacking of discriminatory ability of QC method was the main reason for deficiency. For ER drug product, alcohol dose dumping study is recommended. Some of this deficiency mainly due to partial submission of alcohol dose dumping or increase of alcohol drug release from the test compared to the reference drug. 17 deficiency was related to the pharmacokinetic. Some ANDA exclude subject without proper justification or document, and missing inadequate information on the failed pilot study, especially in study report, and incomplete SAS transporter file was the main theme for this deficiency. 35 deficiency were related to the bioanalysis. Missing analytical raw data and chromatogram and inadequate sample reanalysis was the main reason for this deficiency. And missing SOP and validation and also stability data was another deficiency for bioanalysis section. For formulation, exceeded excipient level and missing information on colorant, flavor, and shell was the common deficiency. So the 157 ANDA was sorted based on the receive date, which is pre-GDF, GDF 1, 2, 3. And then I look at how many ANDA was considered adequate during the first cycle BE and review time. And review time was counted from the FDA receive date to the first complete BE review with the outcome of either BE adequate or inadequate through the CRL. And first cycle BE adequacy rate was significantly increased from the 11% to 83%, and review time reduced 22 to around 7 months as GDF program matures. Case studies about dissolution testing using SCORE tablet for metoprolol-succinate ER tablet. RLD was designed as ER tablet comprised a multi-unit system containing metoprolol-succinate in multitude of the control release pellet, and each pellet acts as a separate drug delivery unit. So as you can see in the picture, the RLD contains multiple pellet, and each pellet can control the drug release. And a film coating is just for easier swallowing, not for functional coating. Therefore, this RLD is a SCORE tablet and can be divided per RLD labeling. So for metoprolol-succinate ER tablet for 100 and 200 milligram, PSG recommend in vivo fasting and fat B study on 200 milligram strength. And additionally, compendial dissolution testing on whole tablet and half tablet is recommended. This is because of RLD, the formulation design, and multimedia dissolution testing and in vitro alcohol dose dumping test recommend in accordance of general guidance. And 100 milligram strength can be waived based on criteria set forth. This is the hypothetical case for metoprolol-succinate ER tablet based on in-house ANDAs. In this hypothetical case, they have two strengths, 100 milligram and 200 milligram strengths. B study conducted on 200 milligram strength on whole tablet and considered acceptable. And dissolution testing, which is alcohol dose dumping and multi-pH media study, were considered and for both strength. And two graphs in the slide are results from the compendial dissolution testing using whole and half tablet. For 200 milligram strength, RLD drug product, half and whole tablet show almost identical dissolution profile. On the other hand, for test drug product, half tablet show much faster drug release compared to the whole tablet. And same trend observed 100 milligram strength. As discussed in previous slide, reference product comprise a multi-unit system. There is no functional coding. So therefore, dissolution profile for half and whole tablet for both strengths are comparable. On the other hand, test drug product showed half tablet is much faster than whole tablet for both strengths. And we think it was related to the product design, test product designed to have drug-loaded matrix in the core and functional coding outside. So as you can imagine, when test product is splitted, the functional coding is going to be compromised. So therefore, additional study were recommended. Applicant was asked to submit fasting and fed study on 200 milligram strength and half tablet, and additionally comparative multi-pH dissolution testing and in vitro alcohol dose dumping testing on half tablet. The reason this study is recommended is because this application has 100 milligram strength too, and alcohol dose dumping and multi-pH media is important for the waiver purpose. Alternatively, it was asked to reformulate test product. As a response to first cycle BE deficiency, the applicant conducted a fasting and fed BE study on half tablet of 200 milligram strength. Both fasting and fed study on half tablet failed because 90% confidence interval of Cmax did not fall within the limit of 80 to 120 and failed in upper limit. So this supports our concern that different formulation design may have impact on bioequivalence of a half tablet. Therefore, applicant was asked to reformulate. Based on this result, we can learn that a good understanding of formulation design of RLD drug product would help to avoid deficiency and reduce the time and effort in drug development process. This slide summarizes my recommendation for avoiding BE deficiency. For dissolution study, please check the PSG and relevant general guidance for identify adequate dissolution testing. Please generate dissolution data using discriminating method. It will be helpful if you submit the method development report or supporting data to show discriminatory ability of your method. Please make sure the stability of test and reference standard product at the time of dissolution testing. For bioanalysis, please submit the 100% analytical numerical raw data, including reject and repeated run, not just for the accepted run, and chromatogram from 20% of serially, not randomly, selected subject. For sample reanalysis, please provide pre-specified reason and proper justification with the supporting document and data. For formulation, please provide quantitative breakdown of the colorant, ink, flavor, and capsule shell. If you need clarification on whether proposed level of inactive ingredient in the test product are acceptable, please submit the control correspondence. For PK, If you exclude the concentration data of subject in BE statistical analysis, please submit the investigation report with the real-time evidence for protocol violation. Please provide SAS data in proper format of data in SAS file, which match with the presented in the BE study report. So all clinical trial studies starting after December 17, 2016 are required to have data submitted in CDISC compliant format, which lists in detailed information in this link. As a summary of my presentation, out of 157 ANDA, 224 deficiency were identified in the first cycle BE assessment for ER drug product, and various deficiency identified during BE review for oral ER product are avoidable. Submitting thorough, well-structured, and acceptable BE data for ANDA will greatly benefit generic drug industry and the American public by facilitating faster and wider availability of generic drug. For today's talk, I'd like to thank DB3 management, April Key for support, and Ligong for initiating this project, and Fangru for helping feedback during clearance process, and OB management, Dr. Bingni and Partha Roy. Thank you for attention.
So hi, everyone. Thank you, Dr. Kim, for the great presentation. So now I'm going to talk about the common clinical deficiencies in ANDAs containing comparative clinical endpoint studies. My name is Ying Fan, and I'm from Division of Clinical Review in Office of Safety and Clinical Evaluation in OGD. So the objective of my talk today first is to understand the goal and the study design of a comparative clinical endpoint bioequivalence study, also known as the CCEP-BE study, and irritation, sensitization, and adhesion study, also known as the ISA studies. And second is to get familiar with the common deficiencies in the CCEP-BE study and ISA studies. And the third is to be aware of the relevant guidances for the CCEP-BE study and ISA studies. So now I'm going to give an overview of the comparative clinical endpoint bioequivalence study. In order for us to understand the concept of the CCEP-BE study, first we need to understand the concept of the generic. So what is the generic? In order for a generic to get approved, you need to demonstrate the pharmaceutical equivalence, bioequivalence, therapeutic equivalence, and need to have the same quality standards to the brand name drug products. So when the generic drug products get approved, it has the same active ingredient, strength, dosage form, and the routes of administration to the brand name drug products. And it is also has absence of the significant difference in the rate and extent to which the active ingredient becomes available at the site of action. And also and it will have the same safety and efficacy to the brand name drug products when used in the indicated population according to the label recommendations. So these standards are the results of the Drug Price Competition and Patent Restoration Act, which is also known as the Hatch-Waxman Amendments in 1984. So in addition to the standards that I mentioned to the previous slide, the generic approval also needs to be based on scientific considerations and minimizing duplicative testing. So what is the ANDA versus the NDA? ANDA is the abbreviated new drug application, and it is the application for approval of a generic drug products, and need to demonstrate bioequivalence and pharmaceutical equivalence, and it relies on FDA findings of safety and efficacy data from NDA. And for the NDA, it is a new drug application, and it is the application for approval of a brand name drug product, and it needs to demonstrate the safety and effectiveness of the drug product. So in terms of the review process for the NDA and ANDA, you can see that animal studies, clinical studies, and bioavailability are replaced by bioequivalence in the ANDA. So what is the bioequivalence? The two drug products will be considered bioequivalent drug products if they are pharmaceutical equivalents or pharmaceutical alternatives whose rate and extent of absorption do not show a significant difference when administered at the same molar dose of the active moiety under similar experimental conditions, either single dose or multiple dose. And for the drug products that are not intended to be absorbed into the bloodstream, bioequivalence may be demonstrated by scientifically valid methods that are expected to detect a significant difference between the drug and the listed drug in the safety and the therapeutic effects. So how to establish bioequivalence then? There are multiple study types to establish bioequivalence, such as the in vitro testing, pharmacokinetic PK study, pharmacodynamic PD study, comparative clinical endpoint bioequivalence study, and the weight of evidence approach. So the comparative clinical endpoint bioequivalence study is the least sensitive, least reproducible of general approach for demonstrated bioequivalence. So what is the comparative clinical endpoint bioequivalence study then? It is a comparative clinical study in humans that can determine the bioequivalence of dosage forms intended to deliver the same active moiety at an equivalent rate and extent to the sites of activity. And it may be applied to dosage forms intended to deliver the active moiety locally, forms that are not intended to be absorbed, or drug products for which traditional PK studies are not feasible. So when to do our comparative clinical endpoint bioequivalence study? And those studies are for those drug products have a negligible systemic uptake, and there is no identified PD measure, and the site of action is local. And examples such as the topical drug products for the skin, nasal spray for the nose, and local acting GI tract products. In terms of the study design, the goal is to demonstrate the two products, generic test.T and the reference products R and B bioequivalence, so that the generic can be substituted for the reference drug product. And the study design, it is a blinded, randomized parallel study, and to use the lowest dose possible to detect more sensitive response to formulation differences, and to show the bioequivalent therapeutic effects of the generic and the reference drug products. And also need to show both the generic drug products and the reference drug products are superior to the effect of placebo. So we need to make sure that the two products have the same clinical effect and the safety profile when given to patients under the same conditions. So in terms of the study design, we need to consider multiple factors, such as the dose, target population, target treatment indication, primary endpoint, assessment, time of assessment, and statistical analysis. And these are the relevant guidances that I would highly recommend that you check before you conduct the study or submit your ANDAs. So the first is a product-specific guidance, we also call it PSG, which have the detailed and specific recommendation for each drug product. And the second is the ANDA submission amendment to the ANDA under GDUFA. It includes the major deficiencies we identify in the ANDA submissions. And the third is the ANDA submission refuse to receive standard guidance, which included refuse to receive deficiencies that we identified in the ANDA submissions. So now I'm going to give an overview of the irritation, sensitization, and adhesion studies. So what is the skin irritation? It is a local arising reaction of the affected skin tissue and appears shortly after stimulation. It is caused by a local inflammatory reaction involving the innate immune system of the skin tissue. Whereas the skin sensitization reaction, it refers to an allergic skin reaction to substance resulting from previous exposure. and it is usually characterized by redness, swelling, and itching. The goal of the ISA study is to demonstrate the irritation, sensitization potential, and adhesion performance of proposed generic transdermal or topical drug delivery system, TDS, is not worse than the reference TDS by using the non-inferiority test. So these are the regulatory history for evaluating the adhesion, irritation, sensitization recommendations. So first, we published the general adhesion guidance in June 2016. And the first revision was published in October 2018, which is together published the new irritation and sensitization guidance. And in April 2023, the second revision of the adhesion guidance and the first revision of the IS guidance were published. And these two guidances give a general study design and the statistical analysis method for adhesion, irritation, and sensitization evaluation recommendations. And the third is the product-specific guidance. After those two general guidances were published in October 2018, so we updated all the TDS PSGs. In terms of the study design, to evaluate the irritation and the sensitization potential, we recommend that you conduct combined irritation and sensitization study. It is an evaluator blinded randomized study, which include 21 days of induction phase, 14 to 17 days of rest period, 5 days of challenge phase, and re-challenge phase if needed. And the treatment arms, we recommend to be marketed generic and reference products. And if it has a safety concern for conduct a normal irritation and sensitization study, for some drug products, we recommend that you use the vehicle TDS and a positive control TDS, which produces mild irritation. And in general, a subject's body movement should not be restricted during the study. And for products with wear period of equal or greater than 24 hours, we recommend that the subjects be permitted to bath or shower routinely during the study in a manner consistent with the label use of the ILD. And the TDS should not be protected from direct exposure to water during such routine activities. And to evaluate the adhesion performance, we recommend you either conduct adhesion alone study or combined adhesion PK study. So it is a single dose randomized 2 treatment 2 period crossover study, and the treatment arms we recommend is the to be marketed generic and reference products, and the duration is the maximum label duration of the wear of the reference product. So in general, the subjects should not be allowed to freely conduct normal activities in the study unit or at home, and may reasonably be expected to occur during the label duration of use for the products. And for the products with wear period up or greater than 24 hours, we recommend the subjects be permitted to bath or shower routinely during the study and in a manner consistent with the label use of ILD, and that the TDS should not be protected from the direct exposure to water during such routine activities. These are the relevant guidances that I would highly recommend that you check before you conduct your ISA study or submit your ANDA. So which include the product-specific guidance, the general guidance for the adhesion, and the general guidance for the irritation and sensitization specifically. And now I'm going to talk about the common deficiencies identified in the review of the CCEP-BE and ISA studies. So this pie chart shows the reasons that we sent out for the easily correctable deficiency ECD or information request IR from 2014 to 2024. And you can see the clarification, justification, missing information, each represents about 46% for all the ECD and IR we send out, and followed by the formulation related issues, which is about 8%. So I'm going to talk about each category one by one, because to understand those ECD or IR is very important for you to prevent the review delay and also to improve the quality of your submission. So for the clarification and justification, it includes study days, rescue information, different from guidance recommendation, and inconsistent information. So for the study days, we would recommend that you provide a study day in addition to the calendar day for the concomitant medication data sets and adverse event data sets. And study day means the day during the study, which you can see on the right side, 2 boxes here represents the day during the study for the 2 data sets. These information are very important for us to evaluate the concomitant medication violation and whether the AE is allowed during the study. So for the concomitant medication, we highly recommend that you submit all the medication the subjects took, especially for those medications that could potentially impact the primary endpoint assessment for your investigational drug product. So for the rescue medication, we know some drug products needs rescue medication. So if there's any subjects took rescue medication during the study, we highly recommend that you provide a separate data sets, and for each rescue medication use, we would highly recommend that you provide a separate listing or separate row. So we would like for you to provide the drug name, dose, the date and time when the drug was taken, and in addition to the calendar date, we also recommend that you provide a study date. So here are the 2 examples that if it's different from the guidance recommendations. So first is if you use a different scales as recommended to the guidance, we recommend that you convert to the scales that guidance recommended and submit a study result. And the second, if the TDS moved before reaching the excessive irritation score or different as you predefined in the protocol, we recommend that you provide explanation when it happens. And the second category is the missing information, which include the missing documents and the missing data sets and data definition files. If there's any subject get pregnant during the study, we recommend that you provide the outcome of the pregnancy and attempts to obtain follow-up information. And if there's any revision that you made for the study protocol, we recommend that you provide all versions, including dates, and also summarize the differences between versions. And we also recommend that you provide IRB approval forms and financial disclosure forms. And for the missing data sets and data definition files, here are some examples that we highly recommend that you provide is the data sets in SARS XPT formats, such as the adverse events, concomitant medications, rescue medications, if applicable, medical history, and reasons for subjects' discontinuation from the study. And also remember to submit the data definition files for all variables. And the third category is the formulation-related issues. We would like for you to provide a formulation for test and placebo and the justification of inactive ingredients if different than the reference listed drug products, and the manufacturing expiration date and lot of batch number for your drug products. And also the clarification on whether to be marketed formulation was used in the studies. So to summarize my talk today, first is to understand the goal and the study design of the CCEPBE study and ISA studies. And the second is to use the product-specific guidance or recommendations. Third, other approach may be acceptable, but require justification. And last but not least is to provide justification, clarification, or other information in original ANDA submission. So here are all the references that I mentioned in my talk. So we can go to the challenge questions. So the first question, to minimize delay of review process of ANDAs that may delay approval of generic drug products, applicants are strongly encouraged to submit all relevant information in the original ANDA submission, including A, clinical study protocol, including all revisions, B, pregnant information and follow-up information if applicable, C, data sets, D is a formulation, E is all of the above. Which one you think is correct? Yes, E is the correct answer. So the second question, which of the following guidances will be helpful to check before conducting irritation, sensitization, and adhesion study? So the first is the general guidance for the irritation and sensitization. Second is the general guidance for the adhesion. C is the product-specific guidance of the study drug products. And D is all of the above. Yes, D, all of the above is the correct answer. So that's all I have today, and thank you for your attention, and I'm happy to answer any questions that you have in the following panel discussion section. So I'm now going to hand over to Rory.
All right, so at this time, we can welcome our previous speakers up to the panel to begin our panel discussion and Q&A. So also, I would like to give a reminder to those in-house, go ahead and line up at our 2 microphones here. They're both by these 2 pillars. If you have a question, go ahead and line up so we can gauge how many questions we have. I'll invite anybody that has questions in-house at this time to go ahead and line up. If we have anybody. And then if not, we can go ahead and begin with some of the, oh, we might have one right here. All right, I'll go ahead and let you go too.
Hi, this question is related to approval timelines, the review cycles. So the communication during cycle 1 is excellent. We love getting the IRs, the DRLs, so we can get the agency the information they need, to continue the review. But then a CRL comes and then it's silence until the end of the second review cycle, and then a CRL shows up. And sometimes it's related and sometimes it's totally different questions. And I'm wondering would it be possible to get some additional communication in that second or subsequent review cycles?
So what I've heard in terms of the question is enhanced communication during the second, third, the subsequent cycles during review, similar to what we're doing in the first cycle review. I think an aspect, that it's great understanding the fact that those additional resources of the staff, they're looking for things at the DRL stage, looking toward those timelines and information. I think we always strive to provide as much, especially through the IRs, as much as possible, and that. I think understanding that, and then as we come to the GDF 4, especially having those as priorities, saying that we want to resource and get that additional, I think would be very, very helpful on everybody's sort of part. But understanding is that, yes, we always want to try to provide as much information, but understanding that with the resources we have, we're trying to use appropriately. I don't know, Diana, did you have, because you're more in the review division and things like that, if you had additional sort of thoughts on that?
Yeah, I think you covered it. It's more of a, we try to communicate as much as we can with IRs, but there's no good of a commitment for DRL in the second and further cycles. So feedback for that going into GDF 4 negotiations and potential, need for increased resources would be helpful, I think.
Thank you.
All right, our next in-house question.
Yeah, my question surrounding discriminating dissolution and exactly what that means to FDA. Do you expect it to fail in the QC media for every parameter or just some parameters? Or can it be out of trend for some of them? And that's very hard to do for an IR product when you just have a single point. So is FDA considering changing like the USP and guidance to say we can have more than one time point on an IR product? And then some of these things you can't get in a controlled correspondence. It seems like FDA is changing their thinking on CBAs and all of that, and all you have is your YouTube videos. And so are you going to change your guidances to actually help direct us on some of these questions?
When you say the one time point, you mean for spec?
Yes.
I review for waiver purpose mainly, but when you show for review purpose, we look at not only one time point, we see multiple time point. We understand why drug products sometimes hard to differentiate, but if you show that we try different formulation and different manufacturing attribute. Sometimes it's not able to differentiate. If you can submit the supporting document, and data, then it will be helpful. But for ER drug product, we generally it is nice to easier to show differentiability of method.
And I would add, this is, we're bioequivalents, at least the two of us here, but this is, biopharmaceutics would have more to say on this, but I think frequently they do hear justification for very fast releasing, immediate release products, why it can't be discriminatory. And so that's something that would be dealt with by biopharm.
I mean, and I understand that, I'm not trying to put you guys on the spot, but when you say discriminating, do you just mean differences? Because we show differences and then we keep getting the same questions, and it feels like they're saying it must fail the QC method. And that's very hard to do, so just to take that back, that, we just need clarification on what you mean by discriminating in a guidance that actually says discriminating dissolution, we mean it must fail, or something like that, because you can show differences in that, but that doesn't seem to be adequate for them. And like you said, for an IR product, it's very hard to do that when you only have one time point. So just something that we need clarification on. Thank you.
All right, thank you for that. And if we have any more in-house questions, I encourage you to step up to the mic at this time. We'll go ahead and switch to an online question, but by all means, please hang up by the microphone and we'll get to you at the next round.
Okay, this question is directed towards Dr. Vivian. How likely is it that low exposure data in a BE study is due to lack of compliance when BE studies are closely monitored with checks of the oral cavity to assure that the study subjects have ingested the drug product? In most cases, low exposure data cannot be verified or confirmed as to the reason for it. Example, subject non-compliance versus formulation failure.
Yeah, it can be hard to say definitively, and that's why the limitations exist for really the AUC that you're seeing has to be below a certain amount. And this is typically for IR products. So this is things that would not be expected to really have formulation issues where they're not releasing. So that was taken into account. This was all internationally negotiated as part of the ICHM 13A guidance. So it really was identified as an issue that is happening, and we are seeing that in some cases there is non-compliance, and we've seen that in the past too with periods where there's just no concentration observed at all. It could be due to maybe there's a mouth tag, but then somebody didn't report vomiting a little bit later. So there could be different things that realistically do happen. And so this is a risk-based approach to kind of identify which of those, based on the product type and based on the data that you're seeing, are likely to actually be due to non-compliance.
Thank you.
Excellent, thank you. We'll take our next in-house question.
I need advice, because in PSG it is given, follow this dissolution as given in the FDA dissolution database. And once we follow that, then the question comes, this is not a discriminating dissolution method. So you generate your own discriminating dissolution method. If that is not acceptable to agency, then why there is a recommendation in the FDA database?
I think that's really good feedback. Maybe we can look at the language of the PSGs if that's an issue where it's not as simple. But I think the PSGs do talk about developing methods if needed.
And maybe also a little bit more clarity. Are you getting these sort of deficiencies in terms of for product quality as to routine QC, or are these a part of like the waivers and things like that?
We have got in both the cases, means, yeah.
Okay, and I do know in terms of, especially from a QC perspective, the dissolution website and database is sort of what they consider as a starting point for you guys to be able to use. It's not necessarily the definitive. And I think what the aspect there is, as you develop your product, we also want to see that you've done due diligence in terms of your development of your dissolution, that being a starting point, providing your rationale and additional information there. But I think as Diana also mentioned, that feedback is also very good for us to basically read back. But I think in terms of for your immediate sort of recommendations is always make sure when you're putting your ANDA together is we start with this, we did this root test, and then providing additional justification and rationale as to why you felt this is fit for purpose as a starting point for that product.
Yeah, thank you so much. This is helpful. Means if we can put that language in the PSG, I think that will help all of us. Thank you.
Thank you for that. We'll take our next in-house question, and by all means, if you have any, please feel free to step up to the microphone at any time. We have time here.
This question is for Kim. So you mentioned we need all strengths dissolution data to get the waiver. So coming to if you have one product has marketing for different flavors. So in that case, so do you recommend 3 lots under the each strength dissolution required?
Usually, yes.
So 3 lots.
3 lots.
Multimedia as well as QC media.
QC, yes. But yeah.
In QC, definitely. Multimedia, we don't, I think, case by case if there's an issue or something, but we don't always recommend. We have to get, Xiaochen says no. One should be okay for multimedia, but
One
QC,
lot.
3, yeah.
Okay, thank you.
Of course, and we'll go ahead and switch over to our next online question, please.
Thank you. This is for Dr. Kozak. For an expedited ANDA, specifically for drug on shortage lists being an IV infusion, what are the expected timelines?
So the question is with regard to a IV drug on drug shortage and expected timelines for the review.
Correct.
I don't have that information off the top of my head. I do know that we have general sort of guidances that talk about the expedited and what those components would be. So I wouldn't necessarily be able to address that question. I don't know if one of my other sort of colleagues would know that sort of process or information. Now, we can get back to you on that, maybe a reply back through another method, but I don't have that information off my head. I don't want to provide advice. I don't have
Okay, thank you. I think Tony here has an answer for us.
In the GDF 3 commitments, regardless of what type of drug product it is, a priority application, if it's a brand new application, will receive an 8-month goal date. If that's the question, that's the way I read it.
Yeah. Thank you.
Excellent. Thank you for that response. We'll go ahead and take our next in-house question here.
Hi, this is for Dr. Diana. So regarding removal of the data, like low exposure. So if we want to correspond to you before submission of ANDA, can we do a control correspondence on it?
Maybe for general questions, if you're asking for pre-acceptance of the data, that may not be appropriate in a control correspondence. You'd have to look at all of the evidence. So I would submit a justification in your ANDA if you already conducted a study. Or if you're wondering what to put in your protocol or something like that, then sure, if it's before the study, you can do a control correspondence.
So post CRL, can we have a meeting on this one otherwise, if we get a CRL on that one?
You can definitely send a control correspondence post-CRL. It depends on if you're asking for clarification. We have the post-CR clarification meetings available. There are certain restrictions for post-CR scientific meetings because there's a longer review time of these are usually more complicated scientific questions. So it depends on what your question is specifically, if it would allow for that.
Thank you.
Of course. And again, just a reminder, anybody in-house with questions, you're here, the mic's here, panelists are here, feel free to step up. We'll go ahead and take a question via online.
Thank you. Addressed to Dr. Kim. For the case study of metoprolol succinate, the score line is functional, but wouldn't that affect the ER properties of the tablets?
For that drug product, as I presented in the presentation, the film coating was just for easier swallowing, not functional coating. So when the even is split it, the ER function is not compromised for that RLD.
Thank you.
Thank you.
Of course. Any takers in-house? Don't think you're safe just because you're behind that other pillar. Anybody? Okay, we'll go ahead and take our next online question then, and we'll keep going until someone steps up.
Okay. For Dr. Fan, can you please briefly explain why the lowest dose should be used in the comparability BE study?
Okay, so the question is if I can provide more explanation about the lowest dose that should be used in the comparative clinical endpoint study. So we treat it case by case. So I would highly recommend that you check that from our product-specific guidance for the recommendation for each drug products. And if you couldn't find product-specific guidance online, for the drug products, that I would highly recommend that you communicate with us in the pre and communication routes, such as the request a meeting or submit control correspondence with your questions. Thank you.
Thank you very much. Let's see. They're trickling in, so have patience with me, please. Let's. Okay, this question I believe is for Dr. Vivian. Is ICH M13 presentation applicable for the sublingual route as well?
If you're seeing this specific issue, it could be, I think generally this route is not covered by ICH M15A survey. Yeah, but if you have this specific issue, like I mentioned, it could be applicable based on scientific principles for other dosage forms also. So you could, come in with some kind of justification why it could apply to your product.
Thank you. Next question, I think, not quite sure who's the most appropriate person to respond, but here's the question. Will ANDA application result in RTR if applicant used its own discriminatory dissolution method and submit data instead of using OGD's dissolution method recommended in PSG?
I can probably take the first stab at that one. As we mentioned, PSGs are our current recommendations and thinking. You can always use an alternative technique and method, but when submitting in the ANDA, provide justification and or rationale as to why you're deviating from our current thinking and recommendations. So long as you provide that additional sort of statement there of, here's the rationale, we're using an alternative method and this sort of aspect, it will, but that usually is what would trigger an RTR if not added. You need to add that additional sort of statement in there that you are using the alternative technique. Then it gets into the review stage. It'll be found at that sort of component if it's acceptable or not. But that's one of the key sort of components. Whenever deviating from a product-specific guidance, please provide that additional information that you are recognizing and that additional rationale. So that way we understand the fact that you are doing a deviation from our current thinking and recommendations.
Thank you. Next question goes to Dr. Vivian. In addition to low exposure data that may indicate subject non-compliance, it's somewhat similar to one we had before, but a little bit different. So in addition to low exposure data that may indicate subject non-compliance, can data be removed from statistical analysis because of real-time documentation of protocol deviations?
Yes, that previous practice is still applicable if there are protocol deviations that were pre-specified that were not met, that your protocol says to exclude because of that, that can still happen. Of course, I think the most clear-cut thing to do is if you observe a protocol deviation like that in real time, you just don't analyze the data, and then there's really no questions.
Thank you. Question for Dr. Kozak, please. If you can you please elaborate a bit more on the nature of facility deficiencies? Also, are these inspection related?
So I'm going to actually defer to our OP colleagues. I believe we're going to talk a little bit later today about that, but I wouldn't have the additional sort of details on what those major manufacturing facilities are at this time.
Okay, thank you. The next question for Dr. Kim, please. Could you share your insights on whether the common deficiencies presented today are specific to ER products or if they are in fact general deficiencies across all oral dosage forms?
I think there are some deficiency related to ER specific, like alcohol dose dumping or multi-pH media dissolution testing, or score, something like that, that is more like ER specific. But I think most of deficiency is common to IR, which I think is relates to the submission quality, which means can avoid it. Thank you.
Thank you.
And just a quick call for any in-house questions. We still have time. The mics are still here.
Mm-hmm. Okay, next online question for Dr. Fan, please. Does FDA have a public list of drug products that for which a comparative clinical endpoint study is required? We realize that for some drug, oh no, I'm sorry.
Okay.
Yeah,
So-
I think.
Okay, so the question is if FDA has a public list for the comparative clinical endpoint by equivalence study is required. So the answer is no, we don't have such a public list. However, we do have the product-specific guidance website that's clearly listing whether the clinical endpoint by equivalence study is recommended or not. So I would highly recommend that you check our website and to see if the clinical endpoint by equivalence study is needed for the drug products that you are interested to develop. Thank you.
Thank you. Okay, this question is for Dr. Kozak. What is the current first cycle approval rate off the top of your head for generics? And can you provide distinction between complex and non-complex products?
Yeah, actually on my graph, I provided at least some first cycle approval rates for both fiscal year 2018, 2022, and I think we're still fairly close. So 2022 and 2020 would be the most recent data. First cycle approval rates are approximately about 20%, and then second cycle about 20%. With respect to complex versus non-complex, we did look at, and off the top of my head, I do have that for complex products, second cycle approval was at least 25%. So we still are seeing as complex products typically require more than 1 to 2 cycles for approval due to the complexity. However, we're still seeing a good 25% within at least second cycle. Obviously, what we can still do is look at more recent sort of things to get that sort of newer thing, especially as we're putting GDF 3 invitation, you get more meetings and things like that. We're hoping and aiming to strive for more first and second cycle approvals.
Thank you. Moving on, next question for Dr. Kim, please. Could you provide some details on control correspondence submission to clarify the acceptability of inactive ingredient level?
Sure. Yeah, you can clarify the acceptability of inactive ingredient through the CC, control correspondence. Briefly, you can submit 3 questions per each control correspondence, like 3 evaluation, and each inactive ingredient and proposed level, each proposed level will count as 1 question. So for example, like 3 inactive ingredients to 1 level, or 1 inactive ingredient with 3 level will count 3 questions. And some drug product indicate for the pediatric, then will have multiple dosing level, then dose range, then each dose range will count as 1 question. So something like this. So, but I think had the control correspondence session was yesterday. If didn't attend, then I think there is a pre-recording, so you can check, and also control correspondence guidance is available. So more detailed information can be found there. Thank you.
Thank you. And for that inquirer, for everyone, the YouTube link from yesterday is on a one-stop website, so you can easily refer to that while it's still fresh. It looks like this is our last online question, and this goes to Dr. Fan. We realize that for some drugs, it is hard to demonstrate the superiority for the treatment arms to the placebo in the comparative clinical endpoint BE studies. Can you give us some recommendations on how best to handle this situation?
Okay, thank you for the question. The question is, we noticed that there are some challenges for demonstrating superiority to the placebo in a comparative clinical endpoint by equivalence study. So we understand that it could be a challenge, and we know sometimes it's very hard to demonstrate the superiority to the placebo for some of the drug products, and it could be due to different reasons, and we handle it case by case right now. So I would highly recommend that you communicate with us as soon as you realize the challenge and issue, and could request a meeting, like submit a meeting request or control correspondence, so we can give you
specific recommendations. And also in the end of submission, we highly recommend that you provide all the information that you think will be helpful for us to review, such as any supporting documents that you think will be valuable, or any literature that information that you think that will be helpful for your application. Thank you.
Thank you. We have one more that's straight in. Hope it didn't lose its way. This is for Dr. Fan. This is regarding transdermal products. Even if we perform PK endpoint study alone, we will measure the adhesion score during PK endpoint study as well. Can we omit the adhesion study?
Can you repeat the question?
Yeah, I hope, I'm reading, so I hope it makes sense. This is regarding transdermal products. Even if we, I think that word is perform, yeah. Even if we perform PK endpoint study alone, we will measure the adhesion score during PK endpoint study as well. Can we omit the adhesion study?
If the purpose is just for the PK, if you want to omit the adhesion study, so the results is going to be just based on the PK. But for the transdermal drug products approval, adhesion is our recommended study that you would have to do to get your generic drug products approval. So if you don't want to do that in the combined PK and adhesion study, you will have to do another adhesion alone study to evaluate the adhesion performance for your drug products. Thank you.
Thank you much. That is all that we have online. Sorry.
All right. Any final last questions here in-house?
I've actually got a real quick plug too. One of my colleagues reminded me that for all those people that are interested actually in the IVBT sort of studies, we're having a Center for Research on Complex Generics, April 29th and 23rd is our next workshop on that. So if you are working on those sort of products, that's another good resource that we've got going. Sorry.
No, awesome. Thank you, Dr. Kozak. And just as another resource, we also do have a selection of guidance documents with QR codes out front. So if anything you heard this morning or anything referred now and you want to have a quick resource, check out that table. You can scan those QR codes with your phone and just take a look at one of the ones. I went through and pulled the ones that were mentioned in these presentations today. If I missed any, my apologies, but hopefully those will be helpful. I think now we are good to go ahead and first take a moment to say thank you to our panelists and presenters today. And from here, we are good to take our break for lunch. We will be back at 12:50. Again, there's a restaurant upstairs. There's also nearby eateries. You can ask the reception desk for more information. And thank you, and we'll see you soon.
All right, welcome back, everybody. I hear the bells going off, so give everybody a few moments just to settle into their chairs. I hope everyone had a nice lunch break, maybe tried something new. So a few more moments, let everybody get settled. All right, so it looks like this is our crowd for the last little quarter that we have of GDF 2025 today. And so we will start with our next session of presenters. First, we will hear from Dr. Jayani Pereira, who is a senior chemist in the Division of Product Quality Assessment. Let's see if I can do this, 19, right off the top there. So in the Office of Product Quality Assessment 3 within the Office of Product Quality within CEDAR. We will also hear from David Green, Senior Pharmaceutical Quality Assessor within the Office of Product Quality Assessment 17 and the Office of Product Quality Assessment 3 within OPQ and the Office of Generic Drugs at CEDAR. Then we will hear from Dr. Feng Wan, Senior Pharmaceutical Scientist in the immediate office and the Office of Product Quality Assessment as one, and then OPQ and CEDAR as well. And last, we'll hear from Andrew Eidzior, chemist in the Office of Pharmaceutical Manufacturing Assessment, or OPMA, within OPQ at CEDAR. We do want to announce right now that our final presenter for this session, Tahsin Mirza, or Dr. Tahsin Mirza, is actually under the weather, so he will not be giving that presentation today. We will skip that portion on the agenda, and we will go ahead and resume and start the Q&A at that time. So you'll notice the agenda is pushed up by about 20 minutes through the rest of the remaining items today. With that, if you will all welcome Dr. Jayani Pereira.
Hi, good afternoon, everyone, and thank you, Corey, for that nice introduction. I hope you all had a great lunch and not too sleepy. If you want to take a nap, don't. So my name is Jayani Pereira, and I'm a senior chemist at Office of Pharmaceutical Quality. And let me go back to one slide. And thank you for the opportunity to present on GDF 3 impact on DMF assessments. Put this down a little bit. Too close to me. So those of you who attended the 2023 GDF may be familiar with most of these slides. We presented it, but this time, we're providing more updated data and also giving you some key points to improve these requests for these GDF 3 enhancements. I'm going to go over some high-level processes, not deep diving into these processes, providing you, like I said, some updated data and key points, and also a couple of key takeaways specifically for the prior assessment process. So let's start with the DMF solicited off-cycle amendments. Seems like this has its own mind and keep going. Hopefully it will stop. So before the GDF 3, most of you remember that we did not pick up any off-cycle reviews. Without an off-cycle means there's no open application. So prior to GDF 3, we did not pick up those solicited amendments. Solicited amendments are any responses to CR letters, deficiency letters. But the one thing to remember is we don't pick up all these solicited amendments in GDF 3. There are criteria we apply. So there will be priority given to these amendments of the DMFs where we can anticipate an approval and result in an approval. So there are a couple of a few criteria we follow. There is a map published and I have a link. I'm sure most of you already read it, and it has actually these priority criteria, the decision tree we apply for these when we triage these amendments. So the high-level implementation process for the off-cycles, so I'm just going to say off-cycles because that's the that one enhancement, the new enhancement in the GDF 3. So when we receive an off-cycle amendment, OPQ, that's us, we triage these amendments based on following that criteria, that decision tree, and we categorize them into two buckets. One is a high priority bucket, the other one is a low priority bucket. So if it falls into the high priority bucket after we going through those criteria, we set a DTD. So the DTD is actually the DMF review target date. So we put a target date and then we assign it to an assessment team. And if there are any deficiencies, we communicate them via IR, information request, if there is no open ANDA at that time. But if there is an open ANDA, then we go ahead and issue the CR. This is actually, without me clicking, it's actually going. I don't know what's going on, so. Oh, okay. Yeah, that's okay. Yeah, when I tested it didn't, but that's okay. I can go back. So bear with me. And if we put any of these solicited amendments into the low criteria bucket, we, depending on the resources, we may pick up them for review, but most of the times they're going to be deferred into until, we receive an application. So this thing jumped right into the slide. It's something really, really important because the trigger for this triaging process is actually receiving this email to the DMF OGD mailbox, which you all are familiar because we had it for a really long time. So this is not something new because in our CR letters, we always say, please let us know when you submit the response because that's a trigger. We triage this mailbox few times a day, every day. So as soon as you send us, oh, I submitted an amendment and we know you submitted it. So that's our trigger. Go ahead and look at it, whether it's off cycle or on cycle. If it's off cycle, we go through the process. So make sure to send that email so that we can do the triaging quickly and assign as soon as possible. So what are the impacts on this enhancement? So like I said, we get an opportunity to review these amendments off cycle. So that means we will have more approval. So Dr. Kosak in his presentation this morning, he shared what first cycle, he said 20% and then second cycle 20%. So that was like prior to GDF 3. So we are hoping that in GDF 3, we can improve that first cycle approvals and also limit the number of cycles for an approval because we are picking up these amendments ahead of time before even an ANDA is submitted. And at the beginning of GDF 3, we estimated about 70 to 100 of these amendments will be qualified under this enhancement. So you can see from this slide that we actually reached that whatever the estimate we made. So we actually in 2023, 76, 2024, 87, and then 2025 up to like February, we've already assigned 22. So we are actually meeting that estimate we made at the beginning of GDF 3. So what this means is that we are finding DMFs adequate quicker than we did before, which actually improves the first cycle approvals and also subsequent like second cycle approvals as well, depending on how many cycles we have. But this is actually a really good improvement we saw when we saw these numbers. So some of the key points for you to know is that we triage these amendments within a week. As soon as we get them, we triage them within a week. If we prioritize under GDF 3, we assign these amendments and we set a DTD 120 days. And you will know that we picked it up for review under this enhancement because we send you an email. Whomever sent that email to us letting us know that they submitted the solicited amendment, we let that person know, or we picked it up for review, and here is a DTD. So the reason to do that is that way, you don't need to wonder whether we picked it up or not. So, but one thing to remember is that if we don't pick it up for review, that means we categorize into the low priority bucket. we don't let you know. But if you wonder, we get so many emails to the DMF OGD mailbox from the industry asking, what's the status of my DMF? So if you wondering what happened, you can always email us and we'll let you know. So that those resources, make sure you use those resources to, communicate with the agency. In some of the slides coming up, I'm going to talk about this communication a lot. So to continue with the key points, a couple of things also you need to know is that once we set this DTD, we don't just change it unless there is a reason, like a valid reason. So one of the reasons to actually change this DTD, so this is off cycle, we assign, but then an application came in with a goal date which requires an earlier due date. So if there's an earlier due date because we need to meet a goal date, we go ahead and change the DTD to that earlier date. The other instances where it would change is, say we picked it up under off cycle, but then all of a sudden there's an unsolicited amendment. So if there's an unsolicited amendment, we can pick it up within the review cycle depending on the type of the amendment, whether it's a minor amendment, but sometimes we have to let you know that it may require a goal date extension. So we ask in that email we send you, just limit the number of unsolicited amendments if you know that solicited amendment is picked up for off-cycle review. So limit the amendments unless you absolutely have to. What if we picked it up for review, we told you what the DTD is, and an application came, but it has a DTD with the goal date at a later date? We don't change it because we made the commitment under GDF 3 to pick up this DMF to review under off cycle, and we meet that DTD. So we don't change it just because there's an application, because now we have an earlier date to commit. Okay, so let's switch into the next enhancement, the DMF review prior to ANDA submissions. We just call them the prior assessments. That's much easier. So this one, again, it's sort of like off cycle because there's no application, but it's called prior assessment because it's prior to a submission of an application. So there are a few criteria the industry needs to follow to be eligible under this prior assessment. One thing is the submission should come 6 months prior to the application submission. So when we talk about this request, the request is actually should be submitted by the DMF holder. It's a request coming from the DMF holder for an anticipated application, which will be submitted in 6 months' time. And like I said, it should be submitted by the DMF holder. And this is available for brand new DMFs, which are not previously reviewed. It could be something you submitted with your prior assessment request, or it's submitted like way, way back years ago, but it has never been reviewed. So those are what we call DMFs without an assessment. So those are brand new DMFs. And there are some few conditions. All of you, I'm pretty sure, already read the GDF 3 commitment, and you already saw that guidance we published. We explained like what type of criteria you should meet. So there are certain criteria you need to meet to be eligible under prior assessment. I just summarized this like a very, it's like 5, I think, but I just summarized it here. Patent and exclusivity expiring within 12 months, no more than 3 approved applications, drug shortage, public health emergency, and sole source product. So those are very clearly outlined in the GDF 3 commitment letter and also the draft guidance. So before you submit anything for a prior assessment, before you submit that request, please read the guidance, please. Guidance has everything. So please read the guidance. There's a checklist for you to say, oh, did I sign the letter? Did we pay the user fee? And did we talk to the applicant? And do we know what the criteria? So there are so many things, and there's a checklist. It's in the appendix. Make sure you read that, get an understanding. And if there's anything confusing, missing, you're not sure, you know where to reach. DMF OGD mailbox. And the prior assessment, there are like two categories, like sort of like two sections. So one is for original ANDAs or ANDA amendments. And then the other one is for prior approval supplements. So the prior approval supplement is sort of limited. It's limited to if you are adding a new API source to a prior approval supplement, which is for your PAS or your application is for a drug shortage or a public health emergency. So this is important because recently we had to deny a couple of these prior assessments, prior assessment request, because there was a submission is for a PAS, but they were not for drug shortage or public health emergency. So because of that, we had to deny it. So make sure, read the guidance. That way you know exactly which category you pick to submit these prior assessments. And here's the link to the guidance. This is the final guidance. It talks about when you can submit it. So for example, prior assessments, you need to have there has to be a DMF. You cannot have drug substance information in the ANDA. They don't qualify. So there are a few things, it's all outlined in the guidance. So make sure, you follow that. Implementation process, just very quickly. So the request comes in and OPQ, FDA, so we triage it. And something unique about this process is we issue this 5-day IR. It's a very short turnaround 5 IR. So it's unfortunately, we cannot send you this IR if you don't have a secure email. Getting a secure email from FDA is very, very easy. So make sure you get a secure email, because if you don't have a secure email, we cannot send you this 5-day IR because it's a short turnaround. We can't fax it or we can't mail it. So let's go through this. So 5-day IR, you need a secure email. We either grant or deny the request. If we grant it, we send the grant letter and we will let you know when it will be reviewed, and we will assign it to a team to conduct the review. And if there are any deficiencies, so most of the times, by the time we finish the review, there is no application in-house. Couple of maybe one time we saw there's an application. So if there's an application, we can issue the CR, but most of the times it's an IR. And if we deny it, we send you a deny letter and we tell you why we denied it. So that's important. We tell you why we denied it so that you can, and also in the deny letter, we tell you, this doesn't mean you denied forever. This means this is the reason why you denied, why we denied your request, so that, you can, if there's something, lack of communication with the applicant, maybe you can, talk to them again, you can submit it again. And we look at it, and then maybe next time you will get a grant. Again, OGD mailbox. So this communication is important. As soon as we see this communication, we look for the project. This is going to be a submission through Gateway. But when we see this email that you submitted a prior assessment, we will look out for that submission. As soon as we see this submission, we're going to start triaging. So we need the notification. Please make sure, send the notification. So what's the impact? Again, have an opportunity to review the DMF outside of an application. Remember that there's a 6 months. Before even ANDA gets into FDA, the DMF is here. So you have that extra 6 months to finish one cycle. So for original ANDA, by the time you come to that mid-cycle, we may have another cycle. So maybe within the first GDUFA cycle, we may have an adequate DMF. So this is why it's very important to, if you are eligible, use this and submit it. The goal is actually, like I said, get to the adequate status as quickly as possible and reduce the number of cycles. And like Dr. Kozak showed, maybe we can improve or increase that percentage of first cycle approvals. So here's the thing. At the beginning of GDUFA 3, we actually estimated we would get about 80. Let's see where we are. We have not seen the numbers. So this is why I am here today again to actually present and remind you this is a really good thing for the industry. Use it. If you have questions, talk to us, ask. Am I eligible? I don't know, like, most of the times it's in the guidance, it's very clear. So use that so you can see, we haven't received that, we haven't seen the numbers yet. So that's why I'm asking you. So make use of it. Key points. Triaged upon request. As soon as we see the email, we look for the submission. So we will triage it as soon as we receive it. We give the industry a chance to fix if something is missing. User fee is not there or there's a user fee issue. Simple things, we send an IR. Grant or deny letter, issue within 2 weeks. If you don't hear from us within 2 weeks after you submit it, ask us what happened to my prior assessment. Send that email. And when granted, we will tell you when it's been done. Usually it is the date you say the ANDA will be submitted. So that's also in the guidance. This is, like I mentioned, for prior approval supplements to add a new source. This is something the industry actually should use for drug shortage and public health emergency. I just put it here for you, but I already talked about this. Planning ahead. Talk with your applicants. Communication is the key. Communication. Talk to the applicant, see whether you are eligible. So that's always really important. I talked about guidance a lot. Yes, follow the guidance. And if you have any questions, email the DMF OGD mailbox. We get tons of emails and we are here to help you get to your first approval, so application approval. So make sure you let us know. Summary, it's exactly the same things that I just summarized. Allow for the assessment outside of a cycle. This will improve the number of approvals. Communication between the holder and the applicant, critical, DMF holder and the applicant, and the communication between even industry and FDA is also critical. We did a really nice, I know I'm out of my time, 15 seconds. We did a really nice workshop back in 2022, right after the GDF 3 implementation. So many resources. We even did a tutorial on how you submit your cover letter. Or you can even submit the 3938 form. So make sure, take use of that and some resources. So I'm out of my time. And thank you so much for listening. And I'll be back later if you have any questions. And with that, I will turn it over to Dr. David Green. Thank you.
Okay, let me adjust here. Thank you, Gianni, for an excellent presentation. It's kind of a tough act to follow. My name is David Green, and today I'm a senior pharmaceutical quality assessor in the Division of Product Quality Assessment 17 in the Office of Product Quality Assessment 3. We're the actives group in the Office of Pharmaceutical Quality. Today I'm going to talk to you about common deficiencies in the drug master files. This is the standard disclaimer that you've seen many times. And here are the mission and vision statements of OPQ and our slogan, One Quality Voice. Thought I'd present that since you haven't seen that. So objectives of today's presentation are first to discuss general considerations regarding type 2 DMFs, the format of the type 2 DMF in the CTD format, and briefly touch on the contents and common deficiencies in each section of the drug substance module. And finally, a brief summary. And I did have challenge questions, but I thought it'd be nice to you and I took them out. So general considerations. First, the type 2 drug master file, the DMF, it deals with the chemistry, manufacturing controls of the active ingredient of a drug product. Reasons for the inadequate, your DMF being inadequate, or the absence of adequate information, which will cause, which usually causes a failure of the completeness assessment, a delay in the DMF becoming adequate, and a delay eventually in the trickle-down thing of the ANDA it's referencing to be approved. It's very important that you understand the critical chemistry manufacturing parameters needed to ensure consistent production of a high-quality API. This goes without saying, provide high-quality submissions. This will ease the review process and get to that first cycle adequacy quicker. And finally, I have a request. In general, this is not a deficiency or anything. It's extremely helpful if applicants submit the 3938 form with their DMS submission. This makes a lot of critical information easily obtained upfront for our staff. The DMF follows the CTT format for the drug substance module. And so there's really 7 sections: the S1, the general properties; S2, the manufacturer section; S3, the characterization; 4, specification; 5, reference standard; 6, the container closure system; and finally 7, the stability. And that's kind of the format of the talk. I will go through section and deficiencies, so forth. So the S1 section comprises of 3 submodules, the first being the nomenclature module. And this is basically every name you can for your substance, the international non-proprietary name, the INN, or the compendial name if applicable, chemical names, other generic names you give it, proprietary names, the CA chemical abstract service registry number, just how you identify. Second subsection is the structure. And again, it's the structure of your API, including the stereochemistry if applicable, the molecular formula and molecular weight. And finally, the general properties section. And this is your physiochemical properties and other relevant properties of your substance. And that can include, and not limited to, physical description, pKa, polymorphism, solubility, characteristics, hygroscopicity, melting point, chirality, isomerism, etc. Common deficiencies we see in this section are missing polymorphic characterization, missing hygroscopicity studies, incomplete solubility data, especially for solvents that are used in the manufacturing process, stereochemical and chiral information about your API, and failure to explain why a certain property is not there. Again, also very common is inconsistent information with known scientific literature. So please make full use of the literature. A lot of this information is out there. The second section of the is the manufacturing section, and it comprises of 6 subsections: the manufacturer information, description of the manufacturing process, control of the materials, control of critical steps and intermediates, process validation or evaluation, and the manufacturing process development section. Common deficiencies in this section are failure to provide all the information on any or additional contractors involved in your manufacturing process, release testing of the drug substance. And that includes the name, the address of the facility, and if there's FEI, DUNS numbers, and so forth, and the responsible person of the facility. It's very critical information that we need. Failure to provide description of any post-processing procedures, such as micronization, how it's performed, equipment used, so forth. This is a big one too: incomplete synthetic schemes. Often we're missing the reagents, catalysts, solvents used, reaction conditions, if intermediates, if they're non-isolated, bracketing. It's very difficult to review without that. And vice versa, incomplete description of the manufacturing process. You have the opportunity to give a description in paragraph form. Oftentimes we're seeing missing batch size, input sizes, theoretical or expected yield counts from each step. Additional deficiencies in this section are lack of adequate justification for starting materials, the designation, missing information on intermediates. And this is intermediates that are, that you purchase from a third-party vendor. And so the full name and address of the manufacturing site and contact information of that vendor, their complete manufacturing information for how they make that intermediate, statements on class 1 solvents for their process, control of impurities and residual solvents in their specifications, the vendor and in-house COA for that intermediate. Finally, a commitment that from the DMF holder, that if the vendor of the intermediate is changed, added, or the process of the manufacturing of the intermediate is modified in any way, that they inform the agency as well as the ANDA holder. And finally, for this section, frequently missing information are reprocessing procedures, solvent recovery processes, critical process parameters and material attributes, complete COAs for all intermediates if they're isolated, and an absence of the process validation or evaluation summary. And again, batch sizes, yield data, and any analytical results is critical. The next section of the application is the characterization section, and that comprises of really 2 subsections, and that's the elucidation of the structure and the impurity section. Common deficiencies in these sections are incomplete characterization of data, including interpretation of the spectral data of the API, failure to address stereochemical features of your API, and these are potential tests you can use: chiral HPLC, specific optical rotation, single crystal X-ray diffractometry, etc. And missing information on the polymorphic characterization of your API. And these are common tests for that. That's PXRD, DCS, FTIR, etc. And finally, and this is very common, unfortunately, poor presentation of the data. It's very hard to assess something if you can't read the copies of your spectra. So it's illegible copies of the spectra are missing missing comparison or expansion information of your data. It's very difficult to, if you can't read it, to know if it's correct. Finally, for this section, missing impurity information. This is new from the last time this is presented many years ago, but M7, failure to provide a complete hazard assessment summary of all potential and actual impurities in this section. We often see missing residual solvents and inorganic impurity assessments, and identification of impurities in a way that we can understand them, or in the sense to provide the IUPAC name and the chemical structure. Sometimes they give company name and no real information other than we can't really interpret what that structure is. So please, it's a very important. Classification of process related, classifying that impurity, whether it's a process or related or a degradant product, and any safety related data that would use as part of your justification if you're justifying the impurity classification. I do want to give a shout out here. It's not on the slide, but this relates to the identification of impurities. If you have the opportunity, please submit the S-structured data file of for your application of all your impurities. It cuts down on a lot of errors that can occur when transferring structures in applications. And a lot of our processes are geared now to really absorb the information out of that file. So please consider submitting that information. Finally, because we've heard a lot about the nitrosamine, but often we don't see the nitrosamine risk assessment that needs to be in this also. And here's the important references in terms of the guidances involved, identifying and controlling all potential nitrosamine small molecules in this case, and nitrosamine drug substance related impurities, and refer to those 2 particular documents. The next section is the controlling of the drug substance, and that's consistent with 5 subsections, the specification, the analytical methods, the validation of analytical methods, batch analysis, and of course, the justification of your specification. And deficiencies we commonly see in this section are incomplete specifications, specifically agreement of your specs with the USB monographs available and ICH Q3A R2. And if there are other impurities that have been listed in other compendial monographs that might not be in the USP, it's good to address them. And whether your unidentified or unspecified impurities meet the ICH Q3A R2 levels. Residual solvents levels, again, limits based on the ICH Q3C, the USP 467, et cetera. Residual metals, if you have a catalyst you use and so forth. And I'm missing a reference here. You should also consider ICH Q3D in that. And finally, quantitative and qualitative counter-ion tests. If you have a particular counter-ion, we often see sometimes that's missing. Also seen in this section are lack of identification tests for counter-ion in terms of the quantitative. This may be requested if the counter-ion is changed during your synthetic process. So keep that in mind. And also missing stereospecific identification tests for chiral drug substances. And there's the reference. Finally, failure to include USP identification tests as part of the drug substance specification. One last slide on these S4 deficiencies. Quantitative results are not reported in actual numerical values. This can be troubling. Failure to control potential mutagenic impurities, and we saw that this goes in hand with the impurity section. And in terms of identification with documentation of the impurity, and again, controlling it per the ICH M7. And M7 has many options to control these impurities. It's not just a limit. And if you do choose to control certain impurities, that higher limits are justified with the pharmacological or toxicology studies. Failure to provide equivalent studies between the in-house and USP method. We often see failure to demonstrate stability indicating criteria of your assay or related substance method. And that it could include no or inappropriate stress conditions, missing peak purity data, missing mass balance between assay and related substance results, representative chromatographs of your method. It's important to provide that as well. Finally, incomplete batch data, and that includes whether the batch data, the batch size, manufacturing date, manufacturing site, et cetera, the purpose of the batch, was it validation or stability. And also failure to provide justification for a specification, maybe based on, but not limited to, your development data, ICH guidelines, batch analysis data, tox data, stability data, USP and other compendial monographs. So the next section of your DMF, it is your reference standard section. There really isn't any subsections to this, so let's go straight into the common deficiencies. First, missing lot numbers of any USP reference standard you might be referencing, failure to qualify an in-house standard against the USP reference standard, missing characterization data for primary standards when there is no USP reference standard available, missing qualification of data for impurity standards. That includes missing spectra, COA, purity, et cetera. And finally, missing identification information for in-house impurity standards that you might be like your working standards and so forth, lot number, source, et cetera. Container closure section is also another section that doesn't really have any subsections. So common deficiencies we see here is failure to provide a certificate for the bag that is in contact with the drug substance. And this is, the reference here is the 21 CFR 177-186 requirements for the indirect food additive regulations and the 21 CFR 177-1520 for LDPE bags. So it's important that that certification is provided. and also your insufficient label information. The label should have, and not be limited to, the name of the API, batch number, storage conditions, retest date, manufacturer's name and address, date of manufacturer, net weight, gross weight, and a caution statement, et cetera. Provide a proper label. The last section of your DMF is the stability section, and that consists of 3 subsections: your stability summary and conclusion section, your post-approval stability protocol and stability commitment section, and your stability data section. Common deficiencies seen in the S7 is failure to provide justification for your stability specifications. Some tests can be excluded, but if they're justified. Failure to support a retest date or expiration date by your stability data. Missing explanation of the out of specification results. In particular, no summary of the investigation. No supporting analytical report for that investigation. And finally, what measures were taken to address the issue that caused the out of spec? Very important information for us. And in summary, as you can see from my slides, some sections generate more deficiencies than others. And that's specifically common for like impurity controls and manufacture and so forth. It's very important to understand the critical chemistry and manufacturing parameters of your API. And I can't emphasize this enough: use regulations, the developmental data, and good science to justify your choices. Make good use of the literature. It's there, use it. Finally, I want to acknowledge my colleagues who helped me with this presentation. And there is no challenge questions, so I let you off the hook. I am going to go ahead now and pass it on to Dr. Fang in OPQA1. So thank you all for your time, and I appreciate you listening.
Good afternoon, everyone. So my name is Fang Yuan from the Office of Product Quality Assessment 1 in OPQ. Let's switch gear from common deficiencies in DMF to the common major deficiencies in ANDAs. So everyone deserves confidence in their next dose of medicine. And our OPQ's mission is to assure pharmaceutical quality that assures the quality medicine to be available and safe and effective. So in this presentation, I'm going to discuss the quality major deficiencies that identified in the first cycle major complete response letters issued in fiscal year 2023. And based on those findings, we'll provide the recommendations to our ANDA applicants to minimize the quality major deficiencies and improve ANDA submission quality, and eventually we can improve the first cycle, second cycle approval rates for ANDAs. So this is a retrospective analysis of the major complete response letters issued in fiscal year 2023 that we conducted last year in CEDAR. And in this study, we collected a total of 284 first cycle major complete response letters. And in those letters, we identified 429 major deficiencies. And based on the issuing assessment disciplines, that 29% of them were issued from the non-quality discipline, and the rest, over 70%, were all quality related. And based on the frequency, the top 3 quality assessment disciplines we identified as manufacturing, specifically facility quality. Majority of them are facility quality related. And the next is drug product, and followed by drug substance. And then we have less than 2% each from the biopharmaceutics and microbiology. So for the major deficiencies issued by the top 3 quality disciplines, manufacturing, drug product, drug substance, we did additional categorization. And based on in accordance with the guidance for amendments to ANDAs under GDF. Actually, last year in September, we just issued the revised version for that. So, so my colleague from the Office of the Pharmaceutical Manufacturing Assessment, Andrew, will discuss the detailed findings in manufacturing-related major deficiencies. And in the rest of my presentation, I'll be focusing on the drug product and drug substance-related major deficiencies. So of the drug product major deficiencies, anyway, so we found that 20% of them were extractable, leachable related, and 20% were related to qualification of impurities. And the 19% were related to drug substance sameness evaluation for complex active ingredient containing drug products. And 14% were related to critical quality attributes either not identified or not controlled. And for those 4 groups of major deficiencies, we did additional breakdown. I'll talk about that shortly. And then 7% of those major deficiencies were due to deficiencies identified through consult for safety assessment or drug device or device design assessment or immunogenic risk assessment. And the rest, 20%, were those major deficiencies that each category has less than 5%. So we group them together under the miscellaneous bucket. So some examples of those major deficiencies include the unacceptable analytical method for quality control. So that as a result, applicant is requested to develop new analytical method. Or unacceptable packaging system that due to inadequate dosing accuracy or potential safety risk. So new batches is needed to using new packaging system to assure adequate product performance and quality. Or if the in some cases, accelerated and intermediate stability studies both failed. So that will require, and the available long-term stability data submitted is not sufficient to establish the expiration dating. And or new drug substance stores is needed. Or the physical properties of the drug product is not acceptable. For example, for our tablets capsule, if the size or dimension is too large, that may raise safety concern. Or in insufficient data to support a drug device compatibility for the proposed product. So extractable leachables related major deficiencies. We found that 75% of them were due to inadequate extractable leachable assessment. So in those cases, even if safety data is submitted, but the drug product assessor cannot facilitate the safety consult to our pharm tox colleagues until those inadequacies identified in the extractable leachable assessment being addressed. And then we have 12% of them were due to lacking safety or toxicology data to qualify the identified leachables above the analytical evaluation threshold. And the rest, 13%, have both type of issues. Unqualified impurities related drug product major deficiencies. we found 52% of them were due to missing QSAR data to assess the potential mutagenic risk of a degradation product in drug product. And 43% of them were due to missing safety data to qualify impurity, qualify the proposed acceptance criteria above the ICH-QT. And 4% of them were due to missing data to assess the immunogenic risk of peptide-related impurities. Drug substance sameness related major deficiencies. So for those ladders, we sampled the drug substance sameness issue were all occurred to the applications for peptide-containing drug products. And we also noted that in those ladders, the application also has deficiencies related to insufficient comparative data of peptide-related impurities profile between the proposed generics and the reference product, either for the purpose of assessing the potential immunogenic risk or for the purpose of establishing quality control strategy for those impurities. So we do have one CMC guidance for peptide containing drug products, though that is only has a limited scope to the peptide synthetic peptide, and thus referencing the product of the recombinant DNA origin. So for the peptide containing for other peptide products that outside the scope of this guidance, applicant may refer to the product-specific guidance. Usually there is recommendation for drug substance sameness evaluation, and also use the proper communication channel like the control correspondence or product development meeting requests to seek the agency's guidance on the expectation for the drug substance sameness evaluation, also and as well as the comparative impurities testing. And also I want to note that the drug substance sameness common deficiencies may also occur to other types of complex active ingredient containing drug products, for example, the polymeric drug substance. So critical quality attributes related major deficiencies, we found that 56% of them were related to insufficient data to address the NDSRI or other nitrosamine impurities risk in drug product. And 90% of them is due to the insufficient data or missing data to demonstrate the physical chemical properties sameness for the complex formulation drug product, usually containing nanomaterials. And 13% of them were due to inadequate nasogastric tube administration studies following to support the label instruction. We do have a presentation this morning discussing those cases. And the rest, 13%, were related to dissolution or control strategy for elemental impurities. So of the drug substance related major deficiencies, we found that 82% of them were due to major deficiency identified in the associated DMF application, DMFs. And 8% of them were due to unqualified impurities. And the rest 10% include inadequate analytical method or insufficient characterization of drug substance or inadequate selection of starting materials, or due to deficiency identified in the Safety Council assessment. So the associated DMF major deficiency letters were also collected and analyzed. And then we found that the 67.5% of them were due to unqualified impurities. and 15% were due to insufficient drug substance characterization, and 7.5% were due to inadequate starting material selection, and others include major drug substance process change or requirement of a new analytical method or requirement of a new drug substance batch. So that is also connected to the David's presentation earlier. So challenging question number 1. Which of the following errors are commonly found with major deficiencies by drug product assessment discipline for ANDAs? A, qualification of impurity. B, evaluation of leachables. C, selection of starting materials. D, all of above. Correct answer is A and B. So we do have 20% found each in the category of unqualified impurities, and 20% related to an extractable leachable assessment. But the selection of the starting material is one of the common major deficiencies in drug substance. So challenging question 2. The most common drug substance-related major deficiency is attributed to unqualified impurities present in drug substance. Is that true or false? Correct answer is true. So we have 8% is due to unqualified impurities, and 82% were due to inadequate major deficiency in the associated DMF. However, in the DMF, we also found the top one common deficiency is unqualified impurities. So altogether, unqualified impurities is the most common drug substance-related major deficiencies. So to conclude my presentation, we want to highlight that manufacturing, especially facility quality, drug product and drug substance are the top quality discipline identifying major deficiencies in the first assessment cycle of ANDAs. And this is same as Dr. Kozak recommended this morning. So FDA and GDF 3 initiatives have enhanced communication with applicants to improve the efficiency of assessment cycle and minimize the common deficiencies. So we highly recommend the applicants to utilize those mechanisms, the communication venues, and also to improve your submission quality and also help with the generic drug development programs. So the key recommendations to minimize quality major deficiencies for ANDA applicants is to ensure facility quality and provide adequate supporting quality data to demonstrate drug substance sameness for complex drug substance containing drug product, and also to support the control strategy for critical quality attributes, including impurities and leachables. And finally, also please strengthen communication with the DMF holders. So I'd like to thank the subject matter experts from the Office of Pharmaceutical Manufacturing Assessment, Office of Product Quality Assessment 1, 2, 3, who helped with the data connection and provided input on the data analysis. And also here, I provided the link to the FDA website for pharmaceutical quality resources, where you can find find a lot of useful information on different topics of pharmaceutical quality. And if you have any general inquiries that is not associated to a specific application, you may submit your questions to this mailbox. With that, thank you all, and I'll see you a bit and hand it over to my colleague Andrew from the Office of Pharmaceutical Manufacturing Assessment.
All right. Hello, I'm Andrew Idzior, and I work as a chemist for the Office of Pharmaceutical Manufacturing Assessment. It's in the Office of Pharmaceutical Quality, and I perform manufacturing process assessments for CMC sections. And I'd like to share with you some common manufacturing process and facility deficiencies associated with ANDAs. So this was designed for the learning objectives and the challenge questions. So we're just intended to allow you to understand some common process deficiencies associated with major withhold recommendations for generic drug applications. And also we'll cover the common pre-approval inspection deficiencies associated with major withhold recommendations for generic drug applications. So we're covering both manufacturing facility related deficiencies associated with ANDAs, but the focus is on deficiencies that supported major withhold decisions for ANDAs. Generic drugs are very important to the US market. They make up 90% of US prescriptions, but it's less than 18% of the total cost. To reach the US generic market, drug applications must go through FDA application process, but a 2019 GAO report found that 12% of ANDAs submitted were approved in the first review cycle. The report considered ANDAs from 3 years, 2015 to 2017. And some years after that report, the FDA has issued almost 300 product-specific guidance documents in the 3 years from 21 to 23. And these guidance documents are intended to help identify the most appropriate methods for developing drugs and generating evidence needed to support ANDA approval. They're available on the FDA website. And a review of the data from the generic drugs program monthly and quarterly activities reports shows that in the 3 years from 2022 to 2024, the percent of ANDAs approved in the first review cycle has increased 50% compared to the amount that was cited in the 2019 GAO report. Although this is progress, the majority of the ANDAs received result in first cycle withhold recommendations and complete response letters. So once a review cycle is complete and an application is found to be not ready for approval, deficiencies are communicated in a complete response letter, CRL. Deficiencies are classified as major or minor based on whether the response to the deficiency will require extensive assessment by FDA. Amendments to ANDAs that address major deficiencies are considered major amendments. Major amendments are expected to require extensive assessment by FDA and longer timelines for such assessment. The review cycles for major amendments can be 6, 8 months, or 10 months. The default is 10 months, but various factors described in the guidance could result in an 8 or a 6-month timeline. However, minor amendments have a review cycle of 3 months. So we just had a challenge question here. The review timeline for amendments in response to major deficiencies is 180 days, 9 months or 18 months, 6, 8 or 10 months, and then 3 months. And we just went over that, so it's 6, 8, or 10 months, of course. Because the FDA assessment timelines are greater for major amendments, the presentation today will focus on the major process and facility deficiencies that were associated with first cycle complete response letters. FDA guidance lists categories of major deficiencies based on the associated assessment team discipline, such as drug substance, drug product, manufacturing process, and facilities. For the process discipline, the guidance documents lists 10 separate examples of major deficiencies. I didn't want to go through all 10 of them. And then for facilities discipline, the guidance document lists 2 examples of major facility deficiencies. They're listed here. A proposed facility is determined to be inadequate, or the ANDA does not include complete identification of the facilities. And we'll continue in more detail about these deficiencies here. To report common process and facility deficiencies, we collected and analyzed data from the first cycle and a complete response letters. The data was limited to ANDAs from fiscal year 2023. From fiscal year 2023, there were 125 ANDAs that received first cycle complete response letters with major deficiencies. The total number of ANDAs for FY23 was about 750. Major process deficiencies were collected as written from complete response letters. Process deficiencies are typically identified as one of the examples listed in the FDA guidance. The major facility deficiencies were all due to a proposed facility determined to be inadequate. From there, additional categorization of the facility deficiencies was performed to describe the reason for those determinations more specifically. So here we have the first cycle major manufacturing process deficiencies from FY23. There were a total of 7 applications within the first cycle that were determined to be not ready for approval due to major process deficiencies. The major process deficiency categories were a need for new exhibit batches to support proposed commercial scale-up plans, a risk of process-related leachable substances that was not adequately addressed by the ANDA, and lastly, a need for new exhibit batches due to a formulation change. So we have another challenge question here. For FY23, only 7 applications were associated with process deficiencies. And then we have a simple true or false. And the answer here is actually false. This question is just designed to highlight the focus on the data that we presented. The focus on this data is for FY23, and we're looking at the major process deficiencies. There were many more than just 7 process deficiencies, but the focus here is on the major. And it's also focused on those that receive first cycle major process deficiencies that resulted in a complete response letter. And then aside from those common process deficiency categories, by analyzing the data by dosage, we can see that all the major process deficiencies were associated with liquid dosage forms. And almost all the deficiencies within the liquid dosage form category were associated with parental drug products. This is the first column there. There are some FDA resources that address these process deficiencies. For the process deficiency category of new batches needed to support commercial scale-up, a review of the product-specific guidance documents could be helpful. The product-specific guidance documents are available on the FDA website, and some of them do include information for appropriate batch sizes for exhibit batches to support ANDA submissions. There is also guidance released in 2014 that recommends exhibit batch sizes for various dosage forms. It's also listed there. For the process deficiency of a risk for product equipment related leachable substances or pearls, you can access a previous SBIA 2024 presentation on the topic. The presentation was recorded and it's available on YouTube. An additional resource is a previous SBIA 2024 presentation on the toxicology aspects of the pearls risk. The slides for the presentation can be accessed on the FDA website. We're going to move on to the common major facility deficiencies. The major facility deficiencies from fiscal year 2023 can all be considered as fitting in the same deficiency category according to that FDA guidance document, which would be that the proposed facility was determined to be inadequate. From there, the deficiencies can be further categorized by the reason for that determination. These reasons include observations made during a GMP surveillance inspection of the facility, observations made during a pre-approval inspection of the facility was actually not ready for an inspection, and quality information was not provided in the ANDA for device components for the manufacturing facility. When we analyze these deficiency categories by dosage form, we can see that only liquid parenteral drug products and solid dosage forms such as tablets, capsules, patches, oral powders were associated with pre-approval major deficiencies in the first review cycle. From the major pre-approval inspection deficiencies, the associated inspection observations were collected and analyzed. The chart here represents 22 ANDAs from fiscal year 2023 that were associated with first cycle major facility deficiencies due to pre-approval inspections. Most of the inspections resulted in 3 observations. However, it's interesting to note that some of the pre-approval inspections only had a single observation. The observations from the pre-approval inspections were categorized into broad categories listed here. Observations were related to raw material testing, post-production activities such as stability testing and reserve samples, data integrity, quality assurance activities such as training and investigations, which excludes OOS investigations, laboratory operations, microbiological contamination, and that includes controls to prevent contamination, lack of microbiological development data, and cleaning and maintenance that doesn't adequately prevent contamination. There was also production activities, which includes a lack of data to support manufacturing steps, inadequate in-process controls, and visual inspection program deficiencies. Lastly, there are facilities and equipment, which includes equipment not qualified or not available, cleaning and maintenance, and water system deficiencies. In this chart, the liquid products are represented by the blue color. The solid drug products are represented by the orange color, and the hash pattern represents the total count. Observations issued by FDA investigators during pre-approval inspections may result in first cycle inadequate major CR letters. Pre-approval inspections of API facilities resulted in first cycle inadequate major CR letters for 4 ANDAs during the fiscal year 2023. They're not represented here. This is focused on the finished product dosage form facilities. Regarding only drug product manufacturing facilities, PAIs resulted in first cycle inadequate major CR letters for 18 ANDAs during fiscal year 2023. You can note that mitigating responses to PA observations may be provided to the FDA before a first cycle assessment is completed. However, the drug product facility inspection deficiencies that are detailed here were not mitigated by such responses. Solid dosage forms, such as tablets, capsules, films, patches, powders, for reconstitution, accounted for 9 ANDAs, and the remaining 9 ANDAs were all for liquid parenteral drug products. So the observation writing style differs for each inspection. Some single observations list multiple deficiencies, while other inspections result in separate observations for each deficiency. Additionally, the significance of a single observation may be considered greater compared to lesser observations. Therefore, entire inspections were associated with categories of deficiencies without actually counting the number of observations for this chart here. The result is that the x-axis of the chart, we have the number of ANDAs that resulted in a first cycle complete response letter for each of these categories. Of the 9 liquid parenteral drug product ANDAs, 7 were associated with microbiological contamination inspection deficiencies, 5 were associated with laboratory inspection deficiencies, and 5 were associated with production inspection deficiencies. And an additional 5 were associated with quality assurance inspection deficiencies. We can see that for the 9 solid dosage form drug product ANDAs, 8 were associated with facilities and equipment deficiencies, 6 were associated with production inspection deficiencies, and 5 were associated with laboratory inspection deficiencies. The categories of drug product manufacturing inspection deficiencies that were most frequently associated with first cycle inadequate CR letters for ANDAs during the 2023 were facilities and equipment, production, and microbiological contamination and laboratory. From there, the inspection observations were further categorized into subcategories to provide more specificity. The table only shows the inspection deficiency subcategories for the liquid parenteral ANDAs. Of the 9 liquid parenteral drug product ANDAs, 5 were associated with inadequate investigations, which actually excludes OOS investigations. 5 were associated with inadequate microbiological controls and procedures, and 5 were associated with lack of microbiological data to support manufacturing steps. These subcategories were all associated with the same number of ANDAs, so there is no clear most common pre-approval inspection deficiency for the liquid parenteral drug products. Moving on to the inspection deficiency subcategories for the solid drug product ANDAs, we can see that from the 9 solid drug product ANDAs, 6 were associated with equipment that wasn't qualified or available. 5 were associated with inadequate in-process controls for manufacturing steps. 4 were associated with inadequate cleaning or maintenance procedures. And lastly, 4 were associated with a lack of data to support manufacturing steps. So it's clear the most common pre-approval inspection deficiency for solid drug products, it's the equipment is not qualified or available. We can summarize the information to identify the top 3 subcategories of pre-approval inspection deficiencies. For liquid parenteral drug products, the top 3 subcategories are inadequate investigations, inadequate microbiological controls and procedures, and lastly, lack of microbiological data to support manufacturing steps. For solid drug products, there was a tie for the 3rd most common category. So listed here are the subcategories: equipment is not qualified or not available. Cleaning or maintenance procedures are inadequate or not followed. Inadequate in-process controls for manufacturing steps. And lastly, a lack of data to support manufacturing steps. We have a last challenge question here for FY23. What was the most frequent pre-approval inspection subcategory associated with process deficiencies for solid dosage forms? And it's actually equipment is not available or qualified. So we're getting close to the end here, and this is a summary of the findings from the data that we collected and analyzed. ANDAs were associated with major process deficiencies. None of them were solid drug product ANDAs. The most common major process deficiency were new batches needed to support commercial scale-up plans and risks associated with pearls. There were 18 ANDAs that were associated with major facility deficiencies from pre-approval inspections of finished drug products. They were evenly split with 9 solid drug product ANDAs and 9 liquid parenteral drug product ANDAs. The most common categories of observations for the liquid parenteral drug products were inadequate investigation, which again excludes OOSs, inadequate microbiological controls, and a lack of microbiological data to support manufacturing steps. For solid drug product ANDAs, the most common observation subcategory was equipment is not qualified or not available. So the data presented here is just limited to FY 2023, and it's just for the first cycle major CRLs for ANDAs. I think we're going to be taking questions as a panel a little bit shortly. And that's all. Thank you.
Yes, and I'll go ahead and welcome our speakers up to join the panel. All right, and as a reminder, just in case you're all kind of sleepy from lunch still a little bit, you're gonna line up here, just like we've been doing these past 2 days at the microphones. For the in-house questions, we'll also switch over to the online questions. If you do have any in-house questions, go ahead and line up, and that way we can kind of gauge what that is. And once we get the all clear from the speakers, we can begin. Let them get all settled. I'm impressed on y'all's timing. That was right on 2:10, so great job.
Wonderful.
All right, we'll go ahead and start with our first in-house question.
Yeah, no pontification this time. Jayani, I wanted to ask you a question. We talk about the prior review of the DMF. Does the inspection also come, like, do you guys also ask the inspectors to do a prior inspection for these DMFs which come as prior, and you accept them for prior review?
Yeah, that's a good question, Aloka, but no, because this is outside of the application, so the inspection, the facilities, those actually come with the application when the application is submitted. So this is only the assessment of the chemistry, manufacturing, and control of the drug substance.
So only when the ANDA is submitted that the inspection will come. Is it okay? I have another small question. David, I was wondering, this is kind of a weird area that, in the DMF guidance says in 32S 2.1, where you name all the facilities, right? And they have said that anything that's involved with the drug substance manufacturing and like release, you should. There are situations where, say, somebody has outsourced an in-process, the raw material and the in-process testing. Now, the raw material we know is not required, but because it's an in-process testing, it's part of manufacturing. Will they have to put that information, you think, in the, at least in the 32S 2.1, where they have the list of all the facilities?
That's a good question, Aloka. Usually the testing facilities that are listed in S1 are S2 deal with the regulatory testing.
Right.
I don't think, disqualified, I would say probably not, but because unless that test is also in a regulatory test further on.
See, that is where the dilemma is, because an in-process test, it's not happening very regularly, but it may be an unusual in-process test, and they are outsourcing it to a lab. I was myself in a dilemma. I thought I'll ask you.
Yeah, it's a good question, and my understanding, it's usually just the regulatory testing facilities that are listed in that section. But definitely provide the data.
Oh, yeah, of course.
So Aloka, is that for in-process?
Yeah.
For API? I think if you don't put it in the 356H, I think it doesn't hurt to put it in the S.2.
That's what I was thinking. I would be very worried to put it in 356H, but S1, S2.1, we could, and then, based on your decision, if it's a critical.
Yeah, certainly put it there. It's not going
to hurt us separately.
It's not going to be detrimental.
Thank you. Thank you.
Excellent. Thank you for that. We'll take our next in-house question, please.
Okay. So my question is about the pre-approval inspections. And I know they're, always scheduled for during the first review cycle. For foreign facilities, are you still giving advance notice? And does that come at, should they hear by the midpoint of the review cycle, or when should we expect to have that happening?
Unfortunately, that's outside the scope of the presentation, and it's best directed to the people that perform those inspections. It's the now called OII, and they're not represented here today.
Okay, thank you.
Thank you for that. We'll go ahead and take an online question at this time.
Okay, first question here is for Dr. Pereira. The DMF prior assessment seems to be underutilized by industry. Are the current requirements to qualify for prior assessment too restrictive? And what can industry do to make better use of this pathway?
Yeah, that's a good question. Yeah, we're also wondering like why the number of submissions or the requests coming into for the prior assessments low. We not really sure, but what we can say is that the GDF 4 is coming. If the industry thinks that there are ways we can add more categories or like relax some of the requirements. I'm not so sure, but I think that's a good time to bring those into the GDF 4 negotiations and see whether we can add more things or like timeline changes, anything. Right now, we are actually not sure. That's why we are actually, we continue to present about these, trying to help the industry. What we think mainly could be that the communication between the applicant and the DMF holders, because the communication has to happen first to see whether there are applications which follow under those categories. So, reading the guidance and see whether they qualify. That's the first thing. Other than that, actually, honestly, we don't know. Thank you.
Thank you much. Next question is for David Green. It's a bit of a long one, so bear with me, please. Under content and format FDA guidance, mentions requirement to submit legible spectra and chromatograms for reference standards and test samples in 3.2.S.4.3 section. Is it sufficient to include spectra and chromatograms for reference and test samples using analytical method validations only? Does ANDA applicant need to also include spectra and chromatograms of reference standards and test samples used in the testing of ANDA batches? Please don't ask me to repeat.
Yeah, that is a long question. I believe they're asking what spectra should they provide, and not only just for the reference standards are also the standards they use for any test. I would suggest if you have a standard that you've used for a particular test, that you provide all of the characterization data as possible. It makes our review easier, and that includes spectra.
Thank you very much. Next question is for Dr. Yuan.
And we have one in-house, Brenda, if we'd like to switch to in-house.
Oh, I'm sorry.
Yes, of course. If you'd like to go ahead and start with your in-house question.
Curveball question number 3. All of the organizations here are here because we want to meet the requirements that are being set up for us to try to meet. But has there been an assessment of the value of all of these requirements in terms of pharmacokinetics or economics, in terms of the reduced accessibility of usable drugs in the United States? That's a curveball question, but an important one.
Well, thank you for your question. I know it's an important question, but it's kind of out of the scope of what we've discussed specifically in this, our section. Feel free to inquire through the agency, and I'm sure someone can address that for you.
Thank you.
I'm sure someday this is going to be asked by other people.
All right, and we would like to take this in-house question over here first, please. Thank you.
Thank you for the presentations. One question regarding the completeness assessment for DMFs and the timeline. So for before pass submission for a new API manufacturer, we noticed that it's a shorter timeline than 6 months that it's listed on the FDA website. So is there an average timeline, how long it takes until it's, the completeness assessment is completed and it's listed on the FDA webpage so that we can better schedule our pass submission?
Even though it's out of this scope of this presentation, I can take that because I'm part of the completeness assessment team at FDA. So yeah, we usually say submit the DMF for completeness assessment at least 6 months prior to the application submission because we want the DMF to be complete and available for reference before the application is submitted. So that's why that 6 months timeline. So I think your question is more about what's the timeline we take. So there is a goal date. There's a metric involved with the completeness assessment. So there is a goal date involved with the completeness assessment. So it's 60 days from the date, the later date of either the fee payment date or the DMF submit date. So sometimes the DMF is submitted earlier, but the fee is not paid until, very close. So we have 60 days, but we usually strive to actually finish the completeness assessment usually within 30 days. And I don't know whether you know the metrics. We actually, we're supposed to meet like 90%, but we are like 99%. So we say, what I would like to, say for this is if you are late submitting a DMF and the application is, coming like in the next month, reach out to us, DMF4GD mailbox, and let us know that it's coming. What can you do? We've expedited CAs to help industry all the time. So we won't say like, no, you're late, but we will do our best to meet it and find the DMF complete. Thank you.
Thank you.
Thank you for that. And we'll go ahead and take our next online question, then get back to the in-house questions. By all means, if you have a question in-house, please go ahead and line up at the microphones as well.
Thank you. Next question to Dr. Yuan, please. Any suggestion on elemental impurity limits not provided in ICH or USB? And another question is on peptide-related impurities. What should be the approach of identification of impurities between 0.1% and 0.5%, since it can get critical to identify at times?
I'll take the first question related to elemental impurities. So the question is asking about elemental impurities outside the scope of ICH, 3D, and USP. So for that, I would say the best way to do it is before you submit, you may use the control correspondence or control correspondence, maybe it's the best way, to communicate with the agency and seek the assessment discipline's guidance on that. For the second question, can you repeat that a little bit?
Certainly. The other question is on peptide-related impurities. What should be the approach of identification of impurities between 0.1% and 0.5%, since it can get critical to identify at times?
Actually, I'm not a subject matter expert for the peptide products, but I think if depending on which peptide product it is, and if it's the peptide products covered by the CMC guidance, you may refer to that guidance. And if the guidance does not address the question, you may also submit control correspondence or pre-IND meeting requests. And for any other peptide products, you can take the same approach.
Thank you.
Thank you. And we can go to our next in-house question at this time.
Thank you. This is also to the DMF division. I just wanted to ask, David, you talked about the impurities and the deficiencies. And at one point, you did talk about literature. And the issue is we are seeing more and more questions these days where people do read, things. You can search something in Google and find it, and they read, and suddenly there is a huge list of impurities saying, have you evaluated this in your or DMF, which actually becomes very difficult. But because, making stats, these are sometimes university papers, so making standards, etc., are extremely difficult. But I was wondering, it's weird that of late we have seen a rise in that. Is there any reason for that?
I think our reviewers have been instructed to give more guidance or justification for their deficiencies, and especially impurities, and especially if there is an impurity that has been identified from the literature. I would just say that you have every right to use literature back to justify that that your synthetic process can't do that and so forth. So it's just trying to give as much information as possible to companies to make the appropriate, so that they control the appropriate impurities for their process.
Thanks. I just wanted to say that you are right, and guilty as charged. I did that in my time too. But the issue is sometimes these drugs are old and well-known drugs, and somebody might have published a general paper, and university research publication sometimes is not always under GMP, never under GMP. It's not always trustworthy. So it would be probably nice if this is looked, if people are using a general paper from JAX or JOC to cite a big list of impurities for a known product, it may be a good idea to have a look.
Thank you for that. We'll go ahead and take our next online question.
Okay, this question is for Andrew. Recently, the FDA review letters include requests for documents supportive of A, food industry quality as per CFR for polymeric manufacturing equipment, and also B, ASTM certified quality for stainless steel manufacturing equipment. Would this be because of PAI observations?
No, it's not. It's an assessment of the manufacturing process, and the concern is the equipment that's being used for the manufacturing process. They're taking a look to make sure that it's certified for its intended use, and that there's no risk of contaminants, or that it's appropriate for the drug manufacturing process. It's not related to the pre-approval inspections.
Thank you very much.
And I think we have time for one more question from online.
Okay, one more. Who's the lucky person? Let's see. Dr. Pereira, is Model 2.3.S QoS still required for initial Type 2 DMF submission? Will the missing of this section affect DMF completeness assessment?
Yeah, that's a good question. Even the reviewers sometimes ask me. Actually, it's not required, but it's highly recommended. Having said that, no, not having it in the DMF submission is not going to be, it's not going to make the DMF incomplete. So it's recommended, but not required. Thank you for that question.
Thank you for that answer. That's it.
Okay, I think that will be it for now. We're coming right up on time right now. So at this point, we will take a small break, be about a 20-minute break. So at 2:50, if you will rejoin us back in this room for our last of the sessions for this afternoon, and then we'll close out, but I'll let you guys go. And yeah.
had the bells chime, let people kind of settle in for a few more moments. I see some people kind of lingering. Maybe so one more session for the day. So a few more. Okay, so we will go ahead and begin our final session for GDF 2025. For these next three presenters here, our first presenter will be Dr. Truong Quoc, team lead of the Division of Orange Book and Regulatory Assessment within the OGDP within OGD at CEDAR. Next, we will hear from Dr. Xiaozhang Jing, Deputy Division Director in the Division of Bioequivalence 2 within the Office of Bioequivalence within OGD at CEDAR. And then lastly, we will hear from Dr. Feng Liu, team leader in the Division of Bioequivalence 1, within the Office of Bioequivalence in OGD at CEDAR. So if you will please take a moment to introduce with me Dr. Truong Quoc.
All right, thank you, folks. My topic here is actually on a lighter sense compared to others, but today I'll be talking about the Orange Book, mostly the drug marketing status that is available in the Orange Book. So today I would like to present a brief history about the Orange Book, describe the basics of marketing status changes, describe 506I marketing status report requirements, discuss three case studies on 506I marketing, and then present you with a couple of challenge questions. So just to give you a bit background about the Orange Book. The Orange Book was distributed as a proposal in January 1979, and it includes currently marketed prescription drug products approved under NDAs and ANDAs under the provisions of the 505 of the FD&C Act and the regulations at that time. In 1980, the first Orange Book edition was published. And then in 1984, we have the passage of the Drug Price Competition and Patent Term Restoration Act, otherwise known as the Hatch-Waxman Amendments. This was the first time it was written to statute for approved prescription drug products with therapeutic equivalent evaluations. And then in 1985, in the sixth edition, FDA began identifying discontinued drug products in the prescription drug product list sections in the Orange Book, and that denoted as a caret symbol. And then on to 1987, in our seventh edition, a distinct discontinued drug product list, otherwise known as discontinued section, was created in the Orange Book. And then on top of that, we have our OTC, listing of the OTC and listing of the first patent and exclusivity addendums. The Orange Book is broken into four parts. The first part is the approved prescription drug product with therapeutic equivalence evaluation. The second part has to do with the over-the-counter drugs that are listed that may not be associated with the NDAs or ANDAs. The third part is the drug product with approval under sections 505 of the FD&C, which is managed by the Center of Biologic Evaluation and Research. And for that one, it's only shown in our annual edition. And the last part is a cumulative list of the approved products that have never been marketed, are for exportations, are for military use, or have been discontinued for marketing, and we have not determined if that were withdrawn for sales due to safety or efficacy reasons or any other reasons that has to do with marketing. Just like to point out that in our Orange Book preface, we have two sections that deals with the discontinued section and changes in the Orange Book. In sections 1.11, the Orange Book discontinued drug product list includes drug products that have been discontinued, but does not include products that were withdrawn for safety or effective reason, as confirmed by the Federal Register determination. These determination of whether a product was withdrawn for safety or effective reason made since 1995 and published in the Federal Registers are marked with a footnote to prevent repeated citizen petition requesting for determination for the same product. Drugs are added to this discontinued drug product list when manufacturers notify DOPRA that the products are not being marketed or when other reports administrative actions indicates the status. In sections 1.12, the Orange Book is carefully maintained and regularly updated for accuracy. Applicants are encouraged to notify DOPRA of any changes such as ownership changes in marketing status that may move a product to the discontinued drug product list. Under Section 506IA of the FD&C Act, application holder must notify the FDA 180 days before withdrawing a product from sale, or as soon as possible if the 180-day isn't feasible, but no later than the date of withdrawal. Additionally, under Sections 506IB, requires notification within 180 days of approval if a drug product won't be available for sale within the 180 days of approval. Speaking about the publication and how often this is updated in the Orange Book, information is updated by the website, and it's also available, downloadable as a PDF. Updates are made daily, semi-monthly, monthly, and on an annual basis. So what information is included in these updates? We have our daily website updates that happens in the afternoon, Eastern Standard Time, and we have patent information and generic drug approvals that gets updated daily. Next is we have our semi-monthly website updates. This is done twice a month on a Monday. Also happens in the afternoon. This consists of new drug approvals, ownership changes, marketing status updates, various types of changes due to drug listing information like changes related to therapeutic equivalents, reference listed drug, reference standard, and so forth. And that happens for that current month. Next type of update that you'll see is our monthly data file update, our website update, and our printed update, which is also known as the cumulative supplement updates. This is a full copy of the Orange Book, and this happens at the end of the second week of each month and consists of all the changes that happens in the Orange Book for the prior month. And then lastly, we have our printed annual edition of the Orange Book, which consists of a full copy of all the changes in the Orange Book, which occurs in the prior month. And this includes our Orange Book preface, our drug product lists, addendums, and appendices. And I'd like to note that the Government Printing Office has stopped printing copies, hard copies of our annual editions. So we refer you to our website, which provides you with the current annual editions. And also the software, the Orange Book software that we have is no longer being supported on the new operating system. So the most accurate information that you'll find is actually on the website. Therapeutic equivalent search. And then, so moving on to 506I marketing status report. This came about with the FDA Reauthorization Act of ADARA of 2017, which added the section 506I to the FD&C Act. And this has a few different things that goes under here, marketing status, that relates to withdrawal from sale, drug not available for sale, and also a one-time report on marketing status, which requires the NDA and the ANDA holder to provide a written notification to FDA within 180 days of enactment of ADARA, stating whether the NDA and ANDA holder drugs in the active section of the Orange Book were available for sale, withdrawn, or never been available for sale. We recommend that the NDA and ANDA holder should submit a notification of withdrawal from sale or a drug not available for sale in a letter to the applicable NDA or ANDA filed through the electronic gateway within the required time frame of the section under 506I. And the notification should prominently note that this is under administrative change not available for sale. On the other side, we have the marketing begins or relaunch. Similar to the other marketing status, this you also have to submit through the electronic gateway. And here you would promptly title it as administrative change notification of commercial markets. Please note that, updates made in the response to sponsor requests may be delayed if you title it wrong or title it differently. And most importantly, if you submit it through the annual report, we don't get notifications through there. So please, submit it as those two titles that we request so that we can process your request on time. So moving on to the first type of 506, notification of withdrawal from sale. This is this requires the NDA requires the NDA and ANDA holders to provide a written notification to FDA 180 days prior to withdrawing an approved drug from sale. So there's a list of stuff that we request you to add in the cover letter. The first part is the NDC code, which is the drug that is listed. Please note that for applications that have multiple NDCs, we request you to hold on to those until all NDCs have been discontinued, then submit your notification then. Similarly, for NDAs and ANDA holders, for most NDAs, for NDAs who have brand and authorized generic, please make sure that those have been, the marketing status of those both have been ceased before you submit the notification. We won't discontinue your product until both those criteria have been met. On top of that, we have established name of the drug, proprietary name of the drug if applicable, NDA or ANDA number, the strength of a drug, and then the date on which the drug is expected to be no longer to be available for sale. Note that the FDA generally considers it reasonable for this to be the date on which the application holder will or cease its own distribution of the drug product because it's the date that application holder itself has stopped making the drug product available for sale. And I'll point this point out later in our case study. And then we have reasons for withdrawal, for example, lack of demand, license agreement, supply chain disruptions, or anything related to the commercial launch for the drug product within 180 days. Next is the notification of drug not available for sale. This is under 506(i)(b), which requires the NDA and ANDA holder to provide a written notification to the FDA within 180 days date of approval of the drug if that drug will not be available for sale within 180 days of the date of approval. If the application holder intends to market within the 180 days of the date of approval of the drug, you don't have to submit this notification to us. We'll understand. So similar to the other one, we ask you to have the established name of the drug, the proprietary name of the drug if applicable, NDA or ANDA number, strength of the drug, and of course the date in which the drug will be available for sale if known, and then reasons for not marketing the drug after approval. Okay, and then next one has to do with the application relaunch. FDA recommends that the NDA or ANDA holder notify FDA in a letter to the applicable NDA or ANDA file 30 to 60 days before an anticipated launch date. The application holder should prominently identify the submission as an administrative change, notification of commercial marketing, and then we ask you to include the anticipated launch date of the product and whether the submission of the supplement is required prior to reintroduction of the drug product into the marketplace. Most importantly, if a prior approval supplement is required, please submit that, submit the administrative change or notification commercial marketing notice 1 to 2 months prior to the go date of your PES. If we find that the PES is pending or approved or denied, DOPR will not return the drug to the active section until the PES is approved. On to my first challenge question, which of the following statement is not true about the 506(i) notification? A, notification withdrawal from sales when the application has ceased marketing the product under all relevant NDCs. Notification of withdrawal from sale, NDA holders with both brand and authorized generic drug products should only submit their request when both will cease marketing. Notification of withdrawal from sale and notification of drug not available for sale should be submitted as administrative change, notification of commercial marketing to the NDA. or ANDA and filed through the electronic gateway. And D, notification of drug not available for sale if the application holder intends to market within 180 days of the date of approval of the drug. No notification under this section to the FDA is required. So I'll give you some seconds here to think about that. If you answer C, that is not correct. For notification for not available for sale, you would submit that under the not available for sale. This commercial marketing is only if you want to relaunch your application. So on to my case studies that I presented here. This one has to do with a 506I where they're requesting to move this to the discontinued section. As you see highlighted above, this was submitted in June 14. All the requirements are listed there and they're fine. Only thing here is that if you look at the marketing end date, they're requesting it to be on June 30th of 2025. So even though you submit this in June informing us that you wanted this to be discontinued, we're going to assume that the marketing end date would be June 30th of 2025. So we're going to discontinue it there. So my recommendation here is just hold on to it. If closer to that date, 1 or 2 months, and then just inform us then. My next example is an ANDA submitted to a letter to the agency on 11/30/2024 requesting to return all 4th-strain back to the market, but then they also told us there's a pending PES. So, and then when we checked our EOB, electronic orange book, we noticed that it's already in the active section. But then we looked at our record, we saw that there was no annual report at any source since 2018. So, and no distribution, so we moved it to the discontinued section to avoid any confusions on the market that this is available or not. And then once the PS was approved, we then moved it back to the active section of the Orange Book. So to note here is that, make sure you follow the regulations requirements, sending in, keeping us updated and sending in the information. And then just let you know that, so that we won't get any delays in returning your drug back to active. The third case studies I want to point out here is some ambiguity where the request is to discontinue 2 drugs that was submitted in 2020, early 2025, and then they had, and then the 2 drug itself expires in later months, and then in addition, there was another statement down at the bottom saying that these are still in distribution, but we'll let you know when we want to discontinue it. Well, this kind of information doesn't really help us. So we'd rather you just hold on to it and just tell us, 1 or 2 months before it gets discontinued. Other examples that we've seen are if you're going to relaunch your new approval within 180 days, you don't have to tell us to discontinue that. If you're going to plan to relaunch it within that time frame, no notification is required for that one. Another request is that some of these say that they'll either relaunch or discontinue within a quarterly basis. Well, quarterly to us is like 3 months, 2 months or whatever, so that doesn't really help us. So if you can give us like a date frame, time frame, month to year, that would be more helpful for us so that we can move your product back. I'm gonna go through this second challenge question really quick here. All Orange Book changes are published on a bi-weekly schedule. B, if a PS is required prior to launch, the application holder should notify the FDA 1 to 2 months prior to anticipated approval of the supplement that the application holder is seeking market entry. Application holders have multiple NDCs, some of which are still marketed or have authorized generic that are still being marketed, may request the FDA to discontinue their drug product. And then the last D is the NDA or ANDA holder may submit a notification of marketing status change via email to the Orange Book. Which one is true here? There's only one that's true, which is B. The PS itself, once the, you should notify the FDA 1 or 2 months. A wouldn't be right because we do a daily update, provide you a daily update on the new generic drug application and patents. C wouldn't be right because we would ask you to hold off until all NDCs and authorized generics have been exhausted. And D wouldn't be right because we asked you to submit that to the electronic gateway. You can FYI us to the mailbox so that we can get it too, that's fine, but the official way is sending it through the gateway. And then all my slides are based off of these 3 resources here. So you can always go into the resources here and look up that information. And then what I'd like you to take home today is that the Orange Book reads the marketing status change daily and strives to provide the most up-to-date information through regular updates. To facilitate your processing of your request for marketing status update, application holders should adhere to the requirements for making status updates. Your marketing status request should clearly provide the specific dates in which the strength is being withdrawn from sale or returned to market, and the marketing status change are typically updated and published bi-weekly in electronic Orange Book. And if you have any further questions, you can always reach out to our mailbox. And I'll reserve my question for the panel, and I'll introduce Dr. Liang to the podium. Thank you.
Okay. Good afternoon, everyone. So it's my pleasure to speak with you about case studies of using alternative reference standard in bioequivalent studies. So this is the standard disclaimer. So for my presentation, I will start with the background information, maybe review the definition of reference list drug and reference standard, and the general consideration for agency in select reference standard. And I will discuss the alternative options for reference standard when the reference standard is not available or with limited market distribution. The core of the presentation will focus on some of the case studies drawn from control correspondence submitted to FDA, and I'll finish the presentation with take-home messages. So FDA identified reference list drug and reference standard in the Orange Book. And RD is the listed drug to which the ANDA applicant must show its proposed generic drug is the same with respect to active ingredient, dosage form, route administration, strength, labeling, and condition of use, among other characteristics. Reference standard is the specific drug product selected by FDA that the ANDA applicant must use in conducting any in vivo and in vitro bioequivalent testing required to support approval of its ANDA. So ordinarily, FDA select the highest strength of the RLD as the reference standard. When select a new reference standard, when the RLD is no longer marketed, or the selection will prevent a shortage, or FDA confirmed that the quantity of the RLD in the distribution is not sufficient for testing, generally, FDA will select a previous approved ANDA, which is therapeutically equivalent to the RLD and is the generic market leader. There are some other factors to consider in selecting the new RS. For example, FDA select the ANDA that contains all strengths, which can facilitate the drug development. Or the FDA may consider select the ANDA contain the study strengths per the PSG. So in this slide, you can see in many cases, the IS is the RLD, and in some cases, the IS is the approved ANDA when the RLD is discontinued. So a potential applicant may submit control correspondence to ask FDA to select IS when they encountered the issue like IS was not designated in the Orange Book. IS is moved to the discontinued section, and they found that quantity of the reference standard distribution is so limited and not sufficient for testing. Or if they feel like the current designated IS is not appropriate. So if the agency select a new reference standard, the product generally will remain the reference standard, even if the original reference standard resume market. In this slide, for example of doxorubicin liposome injectable, the LD is the NDA, the IS is the approved ANDA. So what are the options when the IS and LD is not available or with limited availability? The option include using authorized generic version of the LD, use other approved ANDA based on the same LD, use different strands of the reference standard, or using a different dosage form of another LD or IS for the same route of administration based on public available bridging studies conducted by the innovator. For the fixed dose combination product, the option including using individual component of a fixed dose combination drug product when there was supportive evidence. So the applicant can use the other approved ANDA, which was previously designated as a reference standard, when the current designated reference standard is unavailable and the applicant procured the previous IS before IS was discontinued and the product is unexpired. The applicant can use LD instead of currently designated IS when both LD and IS are on the market, or the applicant procured the LD before the LD is discontinued and the product is unexpired. So what is an authorized generic drug? So FDA published the list of authorized generic drug on the website. And the authorized generic drug is the same drug product as the LD under the same NDA. It is therapeutic equivalent to the LD. It may have different color, labeling, packaging, NDC number, but it does not carry the trade name in the labeling. It can be marketed by brand name company or other company with permission. The NDA holder needs to submit information on the annual report to notify marketing of the authorized generic. And then an ANDA applicant with proper documentation may use the authorized generic for the bioequivalent studies. So let's explore the case studies from inquiries in the control correspondence submitted to FDA. The first case is an example of using other strands of the reference standard. The applicant could not obtain the LD and the reference standard of 1,000 milligram oral tablet. They ask whether they can conduct bioequivalent study using 2 times 500 milligram strands of the reference versus 1 times 1,000 milligram of the test product. The FDA confirmed that the current IS and LD of 1,000 milligram strands is not available in the US market, and there's no approved ANDA at the time. And the reference 500 milligram strands has been shown to be bioequivalent to the 1,000 milligram strands of the reference conducted by NDA applicant. Therefore, FDA responds to the applicant that their proposal is acceptable. Case number 2 is an example of using different dosage form for the same route administration. So the LD for 600 milligram oral tablet is not available. So an applicant asks if they can conduct bioequivalent study using 1 600 milligram tablet versus 1 capsule of 400 milligram ANDA1 and 1 capsule of 200 milligram ANDA2, refer to the same LD. So the agency confirmed there's no approved ANDA for 600 milligram oral tablet on the market. Under a different NDA, The 400 milligram AND1 is the designated reference standard for oral capsule. And the NDA holder for tablet and capsule are the same. And also the oral NDA applicant demonstrate the bioequivalence between these two products. Generally, combining two different ANDA as a reference standard is not encouraged. The agency also confirmed that ANDA2 was no longer marketed and moved it to the discontinued session. And we also confirmed that no safety concern for using a higher dose of 1,200 milligram dose in the in vivo study. At the time assessment, we also confirmed that 200 and 300 milligram ANDA1 was approved, but has not been marketed. Therefore, the FDA respond to the applicant that their proposal is not acceptable. We recommend them conducting the bioequivalence study using either 600 milligram tablet test versus 2 times 300 milligram capsule from ANDA1 if that 300 milligram capsule become available in the market. Or they can choose to conduct 2 times 600 milligram tablet test versus 3 times 400 milligram capsule from ANDA1. The third case is about oral sublingual tablets. And the RD for oral sublingual tablet is no longer available. And the applicant asking agency to provide specific guidance on appropriate reference standards. The agency confirmed there's no approved ANDA for the oral sublingual tablets. And there's no Federal Register determination or pending citizen petition regarding reason of withdrawal for the RD. And the bioequivalence of oral sublingual tablets to other approved dosage form was not established. Therefore, the agency responded to applicant that the agency is unable to identify an approved reference standard at this time. So the fourth case is about oral tablet. The applicant asked whether they can conduct the relative bioavailability study between tablets and oral solution to support the bioequivalence because the RD for the oral tablet has been discontinued. So the agency confirmed there's no approved oral solution or other dosage form, and the oral solution manufactured by the applicant is not an FDA-approved drug product, and no scientific basis to bridge tablets to unapproved oral solution. Therefore, the FDA responded to the applicant that their proposed alternative approach is not acceptable, and the agency is unable to identify an appropriate reference standard at this time. Case number 5 is for fixed dose combination product. The RD for the fixed dose combination product has been discontinued. So the applicant asked whether they can conduct bioequivalence study, compare the fixed dose combination oral tablet to co-administrated individual oral tablets. The agency confirmed there's no approved ANDA for fixed dose combination drug product. No determination whether the RD was withdrawn from sale for safety or efficacy reason. The NDA applicant demonstrated the bioequivalence between the fixed dose combination product and the co-administrated individual component. Therefore, FDA responded to the applicant, their proposal is acceptable. They need to submit or refer to a citizen petition seeking a safety or efficacy determination for the listed drug before at the time as any future ANDA submission. For case number 6, it's different from case number 5. The NDA applicant did not conduct any study to compare the fixed dose combination product and the co-administrated individual component. And also the applicant did not provide any information to support the bioequivalence. Therefore, agency respond to the applicant that their proposed alternative approach is not acceptable. The agency is unable to identify any appropriate reference standard at this time. To summarize my presentation, alternative reference that may be used to establish BE and support generic drug approval in some instance. If the ILD is discontinued, the applicant should submit or reference a citizen petition to determine whether the withdrawal of the ILD from sale was for safety and efficacy reason before or at the time of ANDA submission. For any question on the alternative reference standard, contact FDA via control correspondence before conducting the study. And the agency's selection of an alternative reference standard is based on confirmation of the unavailability or limited availability of the current reference standard and relevant scientific consideration. So this slide lists several resources that I use for prepare my presentation. And I would also want to acknowledge my FD colleagues, Dr. Call, she's in the audience and she's also expert for this area, and also Ming, Hongling, and Bing for helping me prepare the slides, and also the Office of Generic Drug Policy and all the Division of Bioequivalence assessor team who assess these control correspondence. Thank you for your time and attention. And I will pass on to Dr. Fan for the next presentation.
Good afternoon again, everyone. After almost 2 days extensive learning from our previous speakers, we'd like to share common bioequivalence information requests, tips for facilitating the review process from BE standpoint, self-introduction. I'm one of the team leaders from the Division of Bioequivalence 1. My daily works are reviewing in vitro and in vivo study data. This presentation is part of commitment for promoting transparency and communication between FDA and ANDA applicants. Number one, we'd like to share the information for commonly observed BE deficiency in IRs. Number two, how to avoid those deficiencies, which could reduce the numbers of the review cycle to achieve BE efficacy. We'd like to present our information as outlined here. Purpose, definition, and functions of IRs. Commonly observed deficiency identified in IRs. Tips for avoiding those pitfalls. Takeaways. Information request is one of the approaches to improve the efficiency of a BE review process. In this presentation, we will emphasize on what are those commonly observed deficiencies in issue IR during 2023 fiscal year, and how to avoid those deficiencies, which may facilitate the approvals. First, let's explain the difference and similarity of CRL, DRL, IR. Commonly response letter will be issued after all related discipline or division complete their assessment within one review cycle. DRL is a letter for possible deficiency identified by review team from one division or one discipline, such as bioccurrence or OPQ. Information request is a letter request further information or clarification of a submit data. A FDA regulatory action means CRL approval, tentative approval, which the assessment clerks stops, whereas DRL and IR are considered communications within the review process, which the assessment clerk is continuous. A IR is seeking information or clarification that allow review team can continually complete its evaluation within the review cycle. For example, reviewer will not be able to run the SAS statistical analysis when the SAS data set is incomplete or missing information. A late cycle IR may also be issued for requesting information or clarification during DRL, CRL amendment review at late stage of a review cycle. We retrospectively assess commonly observed BE deficiency from issue IR during 2023 fiscal year. Out of 665 ANDAs assessed by OB, 16.1% of ANDA were issued BEIR before the midpoint of a review cycle. And 1.6% of ANDAs were issued BELCIR, late cycle IR, after midpoint of a review cycle. We further analyze the deficiency types in issue IRs. So most of issue IRs contain only one deficiency. All issue IR contain classified as a minor deficiency, except one IR classified as a major deficiency due to farm top issue on flavor. In this pie graphic, we'd like to present the BE deficiency list in BEIR, LCIR from an overview perspective. We summarized the BE deficiency type encountered and found that most of deficiencies were identified from in vivo B study, with other deficiencies related to in vitro BE testing, statistical analysis, formulation, and method validation, including but not limited to missing composition table of non-standard FDA meal, missing IVRT testing condition, missing SOPs or breakdown table of flavor. I will further explain the BE deficiency by using the specific case scenario with tips how to avoid these pitfalls. In case number 1, SAS dataset is inconsistent with BE study report. For example, 1,800 samples were analyzed in study report, whereas the SAS dataset only contains 1,700 sample data. Apparently, this is inconsistent. In addition, SAS dataset did not include the actual sampling time and period. So we suggest to verify your data set or BE study report before submission and follow the specific recommendations in related guidance. Another common observed deficiency found in a IR is missing the composition table of ink used in the test formulation. We recommend to provide all quantitative breakdown or DMF number if applicable for colorant, flavors, and inks, and so on. After receive your amendment to DRL, BE review team may issue BE LCIR requesting additional information or clarification of a submit amendment. For example, we ask the shape and pole size of the filter to allow us further evaluate the adequacy of IVRT testing. As the LCIR, late cycle IR, issue at the late stage of a review cycle, we recommend submitting your response as soon as possible, which may facilitate the BE adequacy. In this table, we summarize additional example of BE information request deficiency with the tips that could potentially avoid this deficiency. For example, in vitro dissolution testing date listed as January 1st, 2024, whereas the expiration date of the batch listed as December 31st of 2023. Apparently, it's not acceptable because the dissolution testing conducted on invalid batch. Sometimes we also look at the testing date is as 2024, but the batch expiration date 2004. Apparently, it's a typo. But we still need to confirm issue by issue IR. So we suggest to ensure the accuracy of your summary table versus the BE study report to ensure the consistency. We'd like to highlight some specific considerations that could possibly increase the chance of the first cycle BE adequacy, including but not limited to submit high quality and complete applications. The more review cycles, the longer time to approach to BE adequacy. Application quality and applicants' responsiveness to IR, not only if IRs have a maximized value for a specific application, but also key factors to facilitate approvals. To avoid IR deficiency and improve efficiency of a review process, we recommend to cross-verify BE study report and data before submission. We also, for example, double-check expiration day and testing day. We also found fasting SARS data set submit under FAST study report. So we need to pay attention on that. When respond a IR or LCIR, we suggest providing a complete written response with all supporting data or document by due date. As indicated in IR letter, partial response, incomplete information, fax or email response will not be acceptable. In addition, we recommend clearly state which division you intend to submit so that your response can be properly triaged. Here's the challenging question number 1. What % of ENDA assessed by OB in GDF 3 year 1 will issue IRs, LCIRs asking for clarification, additional information? A, 8%, B, 18%, C, 28%, D, 38%. So as shown in slide number 7, 60.1% of IR plus 1.6% of LCIR equal to 18%. So the correct answer is 18%. Challenging question number 2. In which of following scenarios could a IR be considered to have a maximized value for a particular application? A, providing a complete written response to a IR by the response due date? Yes. B, if resolved, use a IR could lead to BE adequacy in the current assessment cycle? Yes. C, application quality and applicant's responsiveness? Yes. So the correct answer is D, as discussed in slide number 14. So data-driven outcome indicate that out of those ANDAs assessed by OB in GDF 3 year 1, about 18% ANDA were issued BEIR, asking additional information or clarifications. Application quality and applicant's responsiveness to IR are key factors to improve the chance of BE adequacy outcome. Please consider those tips in these presentations. So I'd like to express my sincere appreciation to Office of Backgrounds colleagues, especially Sri, Utpal, Ram, Bing, and Pasa for their help on these presentations. Let's work together. Not only high quality submission from ANDA applications, but also BE assessment team commits to complete high quality review in a timely manner to bring effective and safe generic drug products to American people. Thank you.
Wonderful. Thank you for that presentation. I will now invite our other two speakers up to the stage to get settled for our final Q&A session of the day. Hopefully by now, I think we all know the drill, but just in case you forgot, if you're in-house, you can go ahead and stand up and approach one of the two microphones on either side of the room at this time. Please feel free to go ahead and line up. That helps us gauge how many questions are coming. And then we will go ahead, and if they're good to go, we'll get started. Okay, now we'll take our first in-house question.
Thank you. My question is for Dr. Yang. You mentioned that applicants can submit a citizen's petition to determine if an RLD has been withdrawn for safety or efficacy reasons. And it can be submitted before or at the time of ANDA submission. If a product is PFC eligible, should the applicant submit the citizen's petition at the time of the PFC or at the time of the ANDA submission?
PFC, what is it?
Pre-submission facility correspondence?
I think yes, the answer. So your question is you can submit citizen petition before PLC?
At the time of the PFC submission or at the time of the ANDA?
I think prior to ANDA submission, anytime prior you can submit.
Okay.
Yeah.
Thank you.
You don't have to wait until you submit the PLC or ANDA.
Okay, thank you.
Thank you.
Thank you for that response. Again, any other in-house questions, please feel free to approach the mic now. Looks like we have one.
Hi, question for Dr. Yang. Just a general question. We mentioned cases where FDA did not provide reference standard for the applicants. So just generally, I wonder what happens in that case where the applicant is seeking to develop a new generic product where the RS is not available and FDA would not provide an RS for that development project. I mean, what are the options in that case?
It's a good question, but I think as I presented the example, FDA is not available to identify any approved I feel like that would mean that you have to wait. I don't know, that's all I can say. I also feel like sometimes there's an option of B2. I think at some point, I remember we kind of communicate, said, you may can try B2, but B2 may not be always feasible, which I'm not familiar. That is true. I'm sorry to say that. I don't have a good answer.
I didn't know
Yeah.
there are other options.
I feel like when we say we cannot identify, is that we exhausted our approach to, we couldn't find the answer to, yeah.
Thank you for that response. We'll go ahead and take an online question at this time, and then we'll return back to the in-house ones.
Okay, this question is for Dr. Quash. Does a 505(b)(1) NDA applicant need to certify to a patent listed for 505(b)(2) NDA that has the 505(b)(1) product as listed drug if the 505(b)(1) NDA applicant makes a change to its NDA related to the 505(b)(2) patent?
Thanks for that question, but that's sort of outside of this, my slides here. I recommend you submit that as questions to the Orange Book mailbox, then we can answer you more adequate.
Thank you.
Thank you. We'll go ahead and take our next in-house question.
Steven Sir, I'm an independent consultant. This is my first time here at the forum.
Oh, congratulations.
I've enjoyed it so far. Just want to make sure, did you say the Orange Book is no longer printed? It's fully online?
Yeah, the annual edition through the GPO is not, the hard copy is not available anymore. So another one is just go through our website, which provides you with the annual report, the current one.
Thank you. And did we have one more question over here for the in-house questions? Okay. Then at that time, we'll go ahead and take one online question.
Thank you. The next question is for Dr. Chiang. Can ANDA applicant use authorized generic to perform BE study without submitting controlled correspondence?
I think the answer is yes.
Thank you. Okay, me?
Yes, I think we'll, again, feel free to line up at any time at the mics, but I think we can continue on with the online questions.
Thank you. This question is for Dr. Liu. If your response to an IR contains either gratuitous information not requested by FDA or information that requires a more thorough review, does it have an impact on the preset goal date?
May or may not. For example, you did not provide the breakdown table for subcomponent of a flavor, which is requested by agency. And, but also propose to replacing your original flavor with a new flavor, which may trigger a Farm Talk Council, so that it will have an impact on the goal date. However, on the other hand, you submit missing information for SAS data set, for example. Also provide your own SAS data analysis result, right? You provide SAS data set as complete, then the review team will be able to run the SAS. Run the confirmation statistical analysis, which is the same as yours. So the goal date will not be impacted.
Thank you much. Next question is for Dr. Quash. What if an applicant misses the 180-day from approval timeline to submit a not available for sale notification? Will FDA accept the submission if an applicant notifies later?
Can you repeat that one more time? I just want to make sure.
Sure.
I understand.
What if an applicant misses 180-day from approval timeline to submit a not available for sale notification? Will FDA accept the submission if an applicant notifies later?
That's sort of outside of our scope. For you might want to just email OGD policy for that kind of question.
Okay, thank you.
And it looks like we do have one new in-house question as well.
Hi, thank you for the presentations.
If an ANDA contains multiple strengths and only one strength is being discontinued, should a notification of withdrawal from sale submission be made for the discontinued strength, or do we wait for all strengths to be discontinued?
That would just be that strength that is being discontinued.
Okay, thank you.
Thank you for that response. Any more in-house questions? Any more? Okay, then we'll go and return to online.
Okay, question for Dr. Jiang. If the reference product to be used in the BE study is expired during the duration of the BE study, can testing be performed to confirm that the product is acceptable and meets all approved specifications? Is this approach acceptable? In this example, the RLD is no longer manufactured.
So your question about RD, you mentioned that RD is meeting all the specification. Does that mean the expired batch also meeting the quality attribute or drug product release? Can I interpret as that you're saying that expired batch also meeting all the quality attributes?
Yes, Dr. Zhang confirms that's a correct interpretation.
Yes, so I think in that case, you could submit all your data and justification, and then we'll review from there. I mean, I cannot say yes or no because I don't see the data, right? But I think it's potentially you could submit all the information to the agency to review.
Okay, thank you. Next question, Dr. Liu. How does one avoid the BE-IR deficiencies?
Okay. In short, submit high-quality application and also double-check, verify consistency and complete. I also suggest you refer to my presentation slides 16 to 22 that contain a lot of tips. So that can avoid those IR BE deficiency easily. It's an easy correctable deficiency. Thank you.
Thank you. Dr. Chiang, did you have something to add to that? No, okay. Next question for Dr. Quash. What if the drug product will be available for sale on the 200th day, not 180 day? Still, notification should be submitted for not available for sale. As for relaunch, at least 30 to 60 days prior, another notification has to be submitted. I hope you could interpret that correctly.
Okay, yeah. Regardless of not available for sale or not, you should still submit that information for us so that we can move that application.
Excellent. Next question for Dr. Chiang, please. If RLD has 3 strengths, 25, 50, and 100, and RS is 50 milligrams, will conducting BE versus 50 milligram RS get bio waiver on both higher 100 and lower 25 milligram strengths?
I think it's a good question, but it's very specific. I think we have to look at the drug product, PSG. I don't think I can answer right now, but recommend to submit a control correspondence.
Thank you. Next question for Dr. Quash. When submitting a notification that the drug will be discontinued, we are aligning the end marketing date to the latest expiry date of manufactured product. Will the Orange Book be moving our product to the discontinued section aligned to the end marketing date, or will it be moved to discontinued section at the time of the submission of the notification?
Similar to the example I gave, we'll move it to the discontinued section, whichever date you tell us to. For example, the expiry of the batch or whatsoever, that's when we'll move it. But if you don't let us know and you submit it, we'll assume that that's the date of the cover letter that we'll move it.
Understood. Thank you. Next question for Dr. Jiang, please. What factors does the agency consider when selecting an alternative RS?
As I discussed, I think the approved ANDA, if it contains all the strengths of ILD, it'll be more suitable. And also, if they contains the study strengths as per the PSG, that could be likely be selected as a reference standard.
Okay. Next question for Dr. Quash. Can you comment on the similarity and differences between reporting requirements under 506I and 505C regarding product discontinuations?
So the under 506I, you have withdrawn from sale and drug not available for sale, where we asked you to submit those information based off of withdrawn, or if you are within 180 days of approval. The one-time report, I'm assuming that's what you're asking, is a extra requirement under 506I that we ask you to report if there's any changes to your application. Not related to 506I is the relaunch, which is a marketing requirement, and that has its own sets of requests.
Okay, thank you. Moving on, Dr. Jiang. Can the RLD that the firm previously procured when it was designated as both RLD and reference standard still be used for a bioequivalence study now that the RLD is no longer classified as a reference standard?
The answer is yes.
Thank you. All right, let's see. I think we have a couple more for Dr. Quash. For ANDAs that have been discontinued for many years and have not submitted a notification, should a notification of withdrawal or not for sale be submitted to the ANDA as a paper exercise?
I don't understand what a paper exercise is, but if it's in the active section and it has, and if it's not in distribution, we recommend you submit that 506I for us so that we can discontinue it properly.
Okay, thank you. A couple more. Do we have time? Yeah?
Yes, I think we have time.
Okay. Dr. Quash, will a withdrawal or discontinuation commence market? Okay. Will a withdrawal / discontinuation / commence marketing request be processed if the sponsor submits the request in their annual report?
Like I said in my earlier slide, if it's titled differently or if it's submitted to an annual report, we won't get notified correctly. So that'll delay your request. So we recommend you to submit it accordingly to the title that we recommend.
Okay, and I think this is the last question that I have here for Dr. Quash. Will DOPRA process a return to active request if there is a pending or denied prior approval supplement?
As I mentioned in my earlier slide, if there's a PAS either pending, denied, or CR, we will not return it until that PAS is approved. So we would like to request that you inform us when that PS is about to be approved. That would help us.
Okay, I think we have exhausted all our online questions. Thank you.
Okay, final call for in-house questions. We have a few minutes left, so by all means, take advantage of this opportunity while you're here, while the mics are hot. Anybody? Okay, well then at that time, I'd like to first thank our presenters and our panelists today. So if you'll join me in thanking them. All right, and now at this time, I would like to introduce to the stage Captain Kendra Stewart. Kendra Stewart is the Deputy Director of Operations for the Office of Generic Drugs, whereby she oversees the scientific and regulatory operations. Captain Stewart received her Doctorate of Pharmacy degree from the School of Pharmacy at Florida A&M University and completed her clinical pharmacy residency at the J. Ames Haley Veterans Affairs, or VA Hospital in Tampa, Florida. She is a lifelong public servant and has devoted her career of over 23 years to various scientific and managerial roles in the federal government, specializing in the areas of health informatics, drug policy, and regulatory operation. Please join me in welcoming Captain Kendra Stewart.
Thank you for the warm introduction. Good afternoon, everyone. Again, I am Captain Kendra Stewart. I am OGD's Deputy Director for Operations, and it gives me great pleasure to be able to provide the closing remarks for today's SBIA forum. I also want to start off by thanking the Office of Generic Drugs and OPQ for all the presenters who presented today. And I want to thank you all as well for being here and participating in this event. We hope it was informative and useful for you. Okay. So just as a reminder, and I hope everyone learned a lot today. I learned a lot as well from all of the many questions that were being asked in the room. I just want to remind everyone about our objectives for this event, which were to gain practical regulatory knowledge to enhance your ANDA applications, to streamline the assessment process, and ultimately reduce review cycles. So as a reminder, each of us play an important role in our shared public health mission of ensuring patients have access to safe, quality, and effective generic medications. In particular, the Office of Generic Drugs further demonstrates this commitment through our strategic priorities, which are modernizing our processes, enhancing efficiency, and promoting innovation and excellence in generic drug development and approval. So over the last couple of days, you've seen many, many great presentations from experts across the generic drug program on topics such as labeling, best practices for labeling, utilizing the inactive ingredient database, DMFs, nitrosamines, Orange Book, and so many, many more. Also, as our Dr. Darby Kozak mentioned in his opening remarks, OGD also recently launched a resource page for generic drug applicants, which has on-demand training, presentations, and more to help improve the quality of ANDA submissions and potentially decrease the assessment cycles. So if you haven't been able to check that out, I highly encourage you do so. You'll note on the page that it has a robust amount of resources that we believe will be helpful for industry, and we plan to continue to review the resources on that page and make updates as different topics become more relevant. So again, we encourage you to visit the page. Here's a QR code, but I also note there's a number of stands on different tables, so that information is also readily available on many of your tables. Also of note, in terms of resources, all of the presentations that were shared here today will also be available online after the forum, so you can also access those as needed. So, as we conclude from this generic drug forum, I do just want to emphasize the significance of our collective efforts in improving access to generic drugs. Just an oft-cited statistic that many of us have heard throughout the years is that generic drugs make up more than 90% of prescriptions filled in the US, or that generic drugs save Americans in the US healthcare system more than $1 billion a day. And I think that's more important now than ever. So with this compelling metric in mind, we have to consider the substantial impact of our collaboration. So by working together to submit and assess high quality generic drug applications, we can continue expanding access to safe, effective, and quality generic medications. All of this is made possible by the dedication of our industry stakeholders, FDA staff, and our other partners. And so as we move forward, let's continue to build on that momentum. And together, we can further improve the generic drug development process, address emerging challenges, and ensure that American patients have access to the medications that they need. Wait. Okay, I may have gone too far. Okay. So I just want to thank you again. I also, before I depart and hand it back over to Corey, I also just want to thank this amazing SBIA staff who put together a fantastic event. If we can, please just join me in giving them a brief round of applause. All right. So I really appreciate all their efforts, and I just want to thank you all again for being here. Thank you.
And actually, while I'm here, I do want to take a quick moment. This is not on the agenda, but I do want to invite up to the stage Captain Mary Krimsner. She is the Division Director of the Division of Drug Information, which is my home office here in the Office of Communications. She has a few words, so I will hand off the stage to her before we close out for the day.
Thank you, Corey. This was unplanned. And I'd asked Captain Stoddard to come to the stage as well. You know, this is Captain Stoddard's 15th year for the running, growing, and presenting the SBIA program for the center. She has taken this program from one meeting a year to myriad conferences, webinars, and meetings. It's been an extraordinary journey. We've been on it together for 15 years. And she is, I've never met anyone more committed to training, education, transparency, and public health than Brenda Stoddard. So as we adjourn today, she's never recognized. She's the unsung hero of bringing together industry with SMEs and helping people do a better job, do a more efficient job. So I just want to take a moment to recognize her finally. All the good work that she does. Okay, thanks. Thanks, Claire.
What can I say? Mary's one of the best bosses I ever worked for. Without Mary, this program would not exist. And we would not have had the impact we had on the generic drug industry and indeed the pharmaceutical industry for the past 15 years. She has given this program all the support we could have. Every time we said, let's do this, she was, okay, we can do that, and we can do even more. So I give Mary kudos for support of this program. And it's been a really good ride. Mary, you should do this. And without you guys, you know, we would not have a cause to be here. And that is what has motivated us throughout the years. That is what has propelled us. That is what has kept us sustained, the need that you have for us. And we hope that we have helped you throughout the years in every possible way. So I thank you.
All right, and so maybe a little anticlimactic final notes here, but your certificate of attendance will come in the following next day or so. Within 24 hours, we will send out a thank you email as well as a link to that survey. Again, I really do implore you to please take a moment and fill out that survey, provide as much feedback as possible. If you learned one thing today or yesterday or even 5 years ago, please provide that feedback. It is so imperative as we do our future planning and also help support our mission here at SBIA. And so additionally, with that being said, I do want to take a moment and also acknowledge our technical staff in the back with Resolution Think and Jeff. They keep it all going in our registration desk out front. They make it nice and seamless for us to make sure that we can do this. So if you guys could take a quick moment to thank them as well. And to finalize here as well, the YouTube link will be posted to our one-stop shop there as well, so you can find that live stream link in the interim. And that is all we have today. I appreciate you all for coming out, and we can go ahead and adjourn a bit early today. Thank you.